berkaitan, tidak bisa terpisahkan. Dan hal ini adalah kita sharing tentang plan bidding yang hubungan dengan plan fisiologi, otomatis nanti akan ada dengan biologi tanaman, apalagi biokimia dan biometrik hitung-hitungannya. Gitu. Dan kita tahu bahwa program pemulihan tanaman itu saling kerjasama dengan ilmu-ilmu lain. Dan di program pemulihan tanaman juga akan membahas tentang yield yang mana yang kita fokuskan tuh. Setelah itu adalah tentang goal atau tujuan utama. Bagaimana tentang karakter tanaman sendiri, terutama tentang fenotip, genotip dan hubungan famili. Dan penentuan apakah bisa nggak digunakan sebagai kriteria seleksi untuk hasil tinggi. Otomatis kita akan menghitung nantinya itu adalah variasi atau keragaman genetik dan estimasi dari pewarisan sifat. Berdasarkan ini diadakanlah seleksi bagaimana prediksi-prediksi yang akan digunakan. Setelah itu keputusan untuk mengadakan persilangan, mating decision. Terus berlanjut bagaimana struktur program yang akan didiseminasikan. Dan lagi mengalami atau pentingnya suatu evaluasi bagaimana perbaikan genetik. Apakah ada nggak tentang kemajuan genetik? Atau memang peningkatan tentang variasi genetik. Dan nanti berputar lagi tujuan hasil apa yang kita fokuskan. Misalkan tentang sawit, kelapa sawit ini ya. Ini fokusnya adalah tentang minyak. Di situ ada minyak saturated jenuh dan minyak unsaturated atau tidak jenuh. Nah sekarang orang sudah memikirkan tentang bagaimana meningkatkan minyak tidak jenuh pada sawit, yaitu persilangan antara guinensis yang banyak minyak jenuhnya dengan olivera yang banyak atau tinggi kandungan minyak tidak jenuh. Itu sekarang sudah di lapangan rilis semakin gencar. Itu tentang sawit. Tentang kakao adalah bijinya. Bagaimana kita fokus ke biji, terus kelembutan biji atau kelunakan nanti setelah diolah. Begitu juga kalau kita ingin kopi ya, jadi kopi di biji juga, tapi ini adalah lebih kompleks karena itu adalah tentang aroma. Sehingga nanti itu macam-macam respon lingkungan untuk tanaman kopi. Jadi yang with yield hasil mana yang akan dituju. Nanti setelah itu goal yang kita akan peroleh. Dan kalau kita ingin sorghum, ini berarti dia biji juga green. Atau dia itu batang daun untuk silase, pakan ternak. Atau jus atau nira pada batang, dirubah ke etanol. Nah, itu mungkin sumber dari bioetanol, nanti dari sorghum. Nah, itu bisa juga with yield yang akan kita gunakan. Kalau di tebu, nah, yieldnya itu adalah batang. Diambil niranya juga, ada jus, dan dirubah ke gula. Atau memang batang daun, dia untuk pakan ternak. Dan kasava, ubi kayu, diambil akar. Dan juga bisa juga batang daun untuk pakan ternak, atau bahkan juga ada sekarang itu daun untuk sayur ya. Jadi spesifik kasava untuk sayur ada. Apalagi dikembangkan sekarang sedang mengintroduksi dari Thailand yaitu Unila, yaitu tentang ubi kayu weksi. Jadi dia banyak mengandung amilopektin dibandingkan dengan amilosa. Itu juga start juga. Itulah beberapa contoh-contoh tanaman which yield yang akan kita tuju nantinya itu. Jadi dan bagaimana tentang peran fotosintesis tanaman. Kalau sudah bicara tentang fotosintesis, maka berhubungan dengan fisiologi tanaman. Yang dua hal yang berhubungan adalah respirasi tanaman dan transpirasi tanaman. Jadi saya tidak bicara tentang bagaimana proses fotosintesis, bagaimana proses respirasi atau degenerasi, tapi itulah gambaran bahwa kalau bicara tentang fotosintesis, dua hal transpirasi dan respirasi akan ikut di situ. Nah, disinilah pengalaman-pengalaman saya selama mengukur fotosintesis. Jadi beda-beda pengalaman orang lain, tapi ini pengalaman saya pribadi ya. Jadi mulai tahun 70 itu antara SMA ya atau SMP sampai ke mahasiswa itu, bagaimana cara fotosintesis juga nggak pakai alat ya. Jadi bagaimana ya? Nah, gitu. SMA itu hanya gelombang-gelombang udara ke air ditutup, itulah dia itu CO2 atau gas. Gitu. Terus pertengahan pada saat 1990, yaitu saat saya kuliah di Amerika dan di Jepang, itu sudah mulai menggunakan alat-alat yang sedikit membantu, yaitu Li-Cor 6200 dan 6250. Dan tahun 2000-an itu sampai sekarang 2020 ya atau 2019 kemarin kami sudah menggunakan yang lebih advance lagi, yaitu tentang Li-Cor 6800 yang alatnya ada di sini ya, di belakang ini. Ya. Itu yang pengalaman pribadi ini ya, jadi tidak umum, jadi sangat pribadi sekali. Dan pada awal fotosintesis, yaitu pustaka-pustaka yang saya baca, yaitulah bagaimana model untuk fotosintesis, yaitu dengan gas. Ya, dasarnya jadi bagaimana cahaya dan suhu untuk mengfiksasi CO2 pada kondisi stomata yang terkontrol. Jadi yang kedua pustaka yaitu bagaimana fotosintesis dia, laju fotosintesis itu diduga ya dari beberapa parameter, misalkan klorofil daun, kandungan klorofil, kalau udah tinggi klorofil fotosintesisnya naik. Terus lagi bagaimana absorben rasionya, terus persentase nitrogen di daun, terus lagi bagaimana dia potensial water di daun, level radiasi dan stomata juga. Jadi ini yang sering digunakan ya tahun 1973. Dan yang ketiga itu adalah yang sering dibaca adalah bagaimana metode-metode ya agar agak maju sedikit ini, yaitu bagaimana fotosintesis dan respirasi. Jadi pengukuran melalui CO2, gas CO2. Jadi situ dengan bantuan sensor ya, dan bagaimana pengukuran kelembapan dan juga bagaimana suhunya di situ. Nah, pada saat pertengahan ya itu pengukuran itu penggunaan sudah mulai ada kemajuan. Ini ada pustaka ini adalah penggunaan elektroda oksigen ya untuk itu. Dan di sini adalah bagaimana secara umum bahwa CO2 dan H2O ya, itu adalah proses fotosintesis dengan bantuan energi cahaya. Di sinilah penduga-pendugaannya menggunakan konstanta Planck dan frekuensi. Dan dari daun dimasukkan dalam chamber ya. Terus CO2 dihasilkan dari natrium bikarbonat. Terus langsung O2 di evolve ya. Di sinilah ada semacam persamaan pertama, persamaan kedua, CO2 masuk, air masuk, terus bagaimana O2 keluar. Itulah akan ditangkap oleh elektroda. Terus yang ini yang lebih maju lagi, ini udah masuk ke Li-Cor. Bagaimana gas engine lanjut diukur di keadaan terbuka dan kondisi tertutup, ruang tertutup. Jadi jadi itulah pengamatan pribadi, bagaimana ini kecepatan exchange karbon dioksida diukur, itu bisa juga menjadi aspirasi dan juga menduga juga fotosintesis. Dan Li-Cor inilah bentuk Li-Cor. Ini bentuk Li-Cor yang 6.200 dan 6.250. Ini agak sedikit bagus, tapi saat itu saya makainya itu kalau udah cuaca terang enak, begitu tidak selamanya terang, ada mendung, udah terganggu dia, adjustment-nya itu agak lama sehingga terganggu. Akibatnya saya putuskan untuk mengukur di ruangan, dalam ruangan, jadi di open space dan di room ya, dalam control room itu dengan adanya bantuan lampu itu lebih enak, itu tenang semua adjustment. Kalau di lapangan bagus datanya, tapi adjustment-nya agak lama, 6.200 itu. Jadi juga nggak tahu pengalaman ini apakah memang ya itu. Lah sekarang Li-Cor 6.800. Nah, ini enak di lapangan. Jadi adjustment-nya cepat. Berapa dia cahaya bisa untuk di-adjust ya. Jadi cepat sekali datanya, berapa 1 jam 2 jam sudah cukup untuk berpuluh-puluh sampel yang dicari di situ, yang didapat. Nah, inilah kalau kita bisa menyimpulkan sementara bahwa kalau ada pengukuran fotosintesis itu bisa secara langsung atau tidak langsung. Kalau langsung ya bagaimana produksi oksigen dan CO2 yang ditangkap yang tadi itu ya. Kalau tidak langsung ya peningkatan biomasa. Itu yang sering digunakan selama ini. Ya, bisa bobot basah, bisa bobot kering dan biji. Ini yang ada pengukuran fotosintesis ya, ada langsung dan tidak langsung. Nah, saya tidak membahas banyak tentang ini, yang ingin saya katakan, yang ini saya sharing, yaitu fotosintesis itu hampir mirip, tapi prosesnya memang ada C3, C4, dan ada yang tanaman yang bisa dua-duanya. Bagaimana mengelola CO2. Di sini yang C4 pasti ada bundle seed cell. Kalau C3 nggak ada. Itu yang ini, dan contohnya adalah C3 ya padi, C4 ya sorghum ya kan, dan adalah nanas. Ini hanya ingin beritahuan aja, informasi aja. Inilah fotosintesis yang itu. Nah, yang sekarang yang saya ingin praktekkan yaitu bagaimana posisi fotosintesis dengan alat Li-Cor 6.200 itu saat itu ya, ingin melihat atau mengevaluasi perbedaan varietas dalam kondisi dry, kondisi kering. Ini saya teliti. Dan ini saya tanam di lapangan menggunakan 276 pinat, varietas pinat dari seluruh dunia. Dibagi dua, yaitu 121 varietas adalah termasuk kelompok Valencia dan 155 varietas dalam kelompok Spanish, tipe Spanish. Jadi seperti kita ketahui ada 3, yaitu Spanish, tipe Valencia, dan tipe Virginia, kacang tanah. Jadi kebanyakan memang di Amerika, tipe Virginia itu ada potnya 2 besar-besar bijinya. Kalau Valencia itu potnya itu lebih dari 2 pot dalam lebih dari 2 biji dalam 1 pot. Ya. Terus warnanya itu merah gelap atau merah terang gitu ya. Dark red dan bright red. Kalau Spanish itu ya dia agak kemerahan, reddish ya, kecoklatan itu Spanish. Dan kecil-kecil bijinya. Makasih, Mas. Terus hanya 2 biji per pot. Warnanya yaitu tadi ya, kemerahan dan coklat. Dan Spanish itu ya tinggi kandungan minyaknya, sedangkan Valencia itu agak manis, makanya dia direbus, cukup dimakan, enak dia. Kalau Virginia adalah untuk butter ya, peanut butter. Itu yang ini, jadi ada 276 peanut varietas ya, jadi seluruh dunia. Yang saya gunakan di sini adalah tipe Spanish dan tipe Valencia. Kita tahu bahwa tipe Spanish dan tipe Virginia itu yang sering dilakukan penelitian tentang kondisi kering. Yang jarang adalah Valencia. Itu yang dan menarik lagi bahwa di peanut ini ada tipe yang erect ya, tegak, dan tipe yang runner, tipe yang merambat ya, di tanah, dan tipe yang sedang antara medium ya, antara erect dan runner yang merambat. Itu yang peanut. Jadi kebetulan saat itulah mengukur suhu dan curah hujan. Bagaimana curah hujannya ini cukup rendah ya, sangat-sangat rendah. Suhu berkisar antara 25 dan 32. Jadi ini cocok untuk mendeteksi tentang drug ya, kondisi ya. Ya, inilah kondisi percobaan lapang ya. Jadi terlihat jelas bahwa saat-saat Oktober Desember itulah curah hujannya sangat-sangat rendah. Nah, dengan kondisi tersebut, akibatnya respon tanaman berbeda-beda. Jadi itulah dengan adanya daun-daun mengering atau dieback of leaf. Jadi saya skor mulai 1 sampai 5 di sini. Ya, yang 1 ya diebacknya atau pengeringan daun hanya kecil ya, sedikit ya, ada sekitar di bawah 5. Sedangkan yang skor 2 itu ada hijaunya, ya, tapi dia sudah coklat, kurang dari separuh. Ya. Yang tipe 3 itu dominan kering ya daunnya, sedikit yang hijau daunnya. Sedangkan yang 4 sangat sedikit daun, 1, 2 mungkin ya, yang daun yang kelihatan hijau, di bawah 5. Sedangkan yang skor 5 itulah hampir semuanya coklat. Jadi itu yang tidak toleran ya. Jadi inilah keadaan di lapangan, sangat beragam setiap varietas, yaitu dari 276 varietas. Sangat-sangat beragam dengan adanya scoring dieback of leaf di sini. Nah, running dengan korelasi bagaimana dieback of leaf dengan pot wick, rupanya juga bagus juga. Jadi DPF semakin tinggi skornya, maka dia semakin rendah hasilnya. Inilah korelasi antara dieback of leaf dan yield. Begitu juga bagaimana kondisi yield dengan akar. Bagus juga dia. Jadi semakin dalam akar kacang tanah, maka semakin tinggi juga produksinya. Itu yang ada hubungannya. Kita bisa melihat sangat bagus ini. Nah, dari sekian tadi itu, kolasi itu kami kumpulkan 276 varietas. Saya kelompok-kelompokkan menjadi 3 bagian, yaitu yang sangat sensitif pada kondisi dry, ya, kering, yaitu yang daunnya yang memang coklat atau kering semua, dieback of leaf, ya, skor 45. Terus yang medium dry itu skor 3, ya, 2 ke 3 ya. Dan dry toleran itu skor 1. Dan ada yang hijau, full hijau. Itu ada 3 grup. Ya. Di situ saya pilih perwakilan 3 semua, 3 varietas untuk yang sensitif, 3 varietas untuk yang medium, dan 3 varietas untuk yang tahan kering. Ya, dari 276 varietas. Jadi hanya saya ambil 9. Jadi saya hati-hati sekali, karena ini adalah akan mengevaluasi untuk karakter fisiologi. Ya. Nanti, ya, jadi harus betul-betul cermat dalam memilih ini. Nah, inilah dia itu drug toleran, yaitu dari US 605, US 693, dan PI Introduction 24055447 G114, itu yang drug. Yang medium itu dari Indonesia, ya, perwakilan Lamongan, Kukuh Small Seed, dan PI 240551 Mani Blanco. Yang susceptible itu adalah Banti 17, PI 37405, dan Tainan Shiro Soru. Ini dari Jepang ke Taiwan, ini introduksi situ, ya. Nah, dari ketiga grup varietas tadi, varietas kacang tanah, kita tanam di pot, ya, pot yang 1/2000 Wagner, ini, di dalam vinil net house, jadi rumah plastik, ya. Ada 2 water condition di sini. Saya ngukur dengan PF. Kalau udah PF 1,5, jadi itulah maksimum water holding. Dan itu sebagai kontrol, ya, dianggap 100%. Dan yang PF 3,4, yaitu dianggap 40% dari maksimum water holding. Itu sebagai cekaman kering. Jadi ada 2 sini, ya. Ada 2 aja itu yang terpapar, yaitu sebagai kontrol dan sebagai drug stres. Dan mengukur, setelah itu mengukur photosynthesis dengan Li-Cor 6200, 6250, bukan 9 ini, ya. Salah ini, salah cetak, salah ketik, ya. Terus lagi bagaimana respirasi akar, yang tadi memang ada hubungannya antara akar dan hasil, diukur dengan infrared gas analisis model UR020S. Terus kandungan glukose, itu perwakilan dari asimilat, fotosintat, yaitu dengan espilasi. Kandungan hormon endogenus, ABA, abscisic acid, asam asifat, menggunakan espilasi, lalu dengan ELISA. Terus jadi adalah pengukuran hormon endogenus sitokinin dengan bioassay teknik, kromatografi, lalu di spektrofotometri dengan 665 nanometer gelombang U1100 tipenya. Ya, jadi ini ya yang akan saya gunakan untuk menguji bagaimana antara drop toleran varietas dan yang drop sensitif varietas. Jadi dengan adanya dua kondisi water air ya, yang pF 1,5 sebagai optimum water condition dan pF 3,4 sebagai drop stress. Nah, inilah saya masukkan vinyl net house ya, ini. Jadi inilah sebelah kiri saya ya, sebelah kiri saya ini. Ini adalah drop stress ya. Ini 1/2000 Wagner Pot. Dan sebelah kanan adalah optimum water condition. Nah, di sini. Ini kelihatan jelas bagaimana respon tanaman kacang tanah pada kondisi kering ya. Ini sangat-sangat nyata sekali kondisinya. Nah, ini saya ambil foto dari sekian itu, yaitu yang susceptible adalah yang nomor 126 sini, susceptible ini yaitu daunnya tinggal beberapa ini, walaupun ada beberapa yang tahan, tapi ini termasuk kelompok yang susceptible. Dan yang medium yaitu yang 347 ini medium. Nah, jadi kelihatan juga nih daunnya ada yang hijau, ada yang kering sebagian. Dan yang drop toleran 589. Nah, ini memang jelas kelihatan hijau, daunnya tidak ada yang kering ya. Hanya nomor 5 ini yang ada keringnya. Jadi, inilah kondisi-kondisi yang nantinya akan dicek karakter fisiologi respon pada cekaman kekeringan. Jadi ini yang pot eksperimen ya, dari lapangan diseleksi, dapatlah 3 grup, masing-masing grup ada 3 tanaman, jadi ada 9 tanaman. Nah, di sini waktu akar, ini nampak akar pertama kali, karena drop adalah akar yang terkena dampak pertama. Ini performan dalam sebelah kiri saya, itu adalah kondisi kontrol, ini akarnya hampir tidak ada beda, hanya beberapa ini luar biasa akarnya, kacang tanah, bijinya juga. Namun bagaimana dalam kondisi stres ini, nah ini bijinya juga hampir tidak kelihatan, jadi ini bijinya hanya 1-2 dia. Akarnya juga terjadi variasi, bagaimana dia susceptible, bagaimana dia drug toleran. Ini yang ada bisa membedakan bagaimana tentang kondisi cekaman kekeringan, itu pemaparan akar. Nah, dari sini kelihatan bahwa bagaimana aktivitas respirasi akar. Nah, ini drop toleran ini tinggi dia respirasinya dibandingkan dengan yang susceptible. Ini juga kelihatan tadi dari bentuk performance. Jadi itu ya tentang medium itu yang tadi memang dia namanya medium ya, ini bisa tengah-tengah jadi. Jadi yang jelas di sini adalah bagaimana varietas yang tahan kering menambahkan aktivitas respirasi akar yang lebih tinggi dibandingkan dengan respirasi akar pada tanaman kacang tanah yang susceptible pada kekeringan. Bagaimana tentang fotosintesis, laju fotosintesis? Juga gitu ya. Jadi yang dilihat sini dalam keadaan water optimum condition ya, optimum ini yang hitam transparan ini sedikit variasi ya, ini ada yang sama, ada yang beda, tapi yang jelas ini dia dalam kondisi ya. hampir ada yang sama, yang ini ada yang sama dengan yang di drug toleran, di keadaan normal ya. Begitu dalam kondisi drug, nah ini kelihatan sekali bahwa drug toleran ini menampakkan fotosintesis laju fotosintesis yang lebih tinggi dibandingkan yang susceptible. Terutama kita lihat bahwa US 605 ini yang paling tinggi dia. Ini yang jagoan nanti bagaimana performance US 605 dia. Ini yang tahan pada kondisi kering. Ini adalah laju fotosintesis. Datang bahwa sekali lagi bahwa yang varietas tahan kering menunjukkan laju fotosintesis yang lebih tinggi dibandingkan yang susceptible, tidak tahan kering. Ini dia itu. Terus bagaimana dengan transpirasi? Sama ya, transpirasi juga gitu bahwa dalam kondisi optimum hampir sama ya dia. Begitu dalam kondisi drug, lah dia sedikit berbeda. Ini lagi US 605 ini menunjukkan sedikit transpirasi dibandingkan yang susceptible one, yang drug susceptible, yang sensitif drug. Ini sangat tinggi ya di laju transpirasi. Jadi dilihat di sini bahwa ada apa sebenarnya yang di drug toleran ini ya bisa mengontrol laju transpirasi dibandingkan dengan drug susceptible. Yang menarik sekali di sini bagaimana penambahan laju transpirasi. Ini diukur dengan Li-Cor 6.200 saat itu. Nah, akhirnya saya membuat korelasi bagaimana korelasi antara panjang akar, bobot kering akar, juga bobot biji, bobot polong ya. Jadi hampir semuanya adalah nyata ya di sini. Jadi sangat nyata dia korelasinya. Ini korelasi yang sangat menarik. Panjang akar, bobot akar kering juga, bagaimana respirasi akar, bagaimana water use efisiensi penggunaan air, bagaimana tentang leaf chlorophyll juga, bagaimana tentang stem dry weight ya, tentang batang, protein batang, dan pot width atau yield, berkorelasi nyata dan positif. Nah, sekarang bagaimana partitioning fotosintat, yaitu yang saya analisis adalah di sini menggunakan tentang kandungan glukos di daun, di akar, dan di pot. Jadi yang di pot ini yang warna putih, ya. Ini yang kandungan glukosa. Terus yang di root, maaf ya, yang di root ini akar ya, bukan di pot. Yang di akar ini yang warna putih. Yang di arsir itu adalah yang di pot cell ya, saya maksudkan ada apa ya, cangkang ini ya, kulit pot. Yang di daun ada yang transparan, apa ya, hitam transparan, ini yang di daun. Jadi ini di daun, yang arsir yang transparan dan transparan, yang di arsir adalah itu di pot, yang di akar adalah putih. Itulah ya kandungan glukosa. Jadi sedikit beda di glukosa sini, terutama ini ya, terutama yang menarik adalah daun. Ini memang daun ya, kandungan glukosa tinggi dia dibandingkan yang di drug sensitif. Jadi itu yang yang terpapar benar ya, terpapar. Jadi ini dalam kondisi drug, saya ngamati bagaimana partitioning glukosa dari daun, akar, dan pot. Lalu saya buat korelasi bagaimana panjang akar dengan pot width. dalam kondisi stres. Nah, bagus juga dia adalah positif nyata operasinya. Begitu juga bagaimana tentang lip greenness, yaitu dengan mengukur spot dengan lip fotosintesis. Ada ya, jadi bagaimana kehijauan daun dengan laju fotosintesis. Ada juga hubungannya di sini. Lalu bagaimana hubungannya dengan fotosintesis dan pot width? Nah, di sini tadi sudah terjawab sebagian bahwa fotosintesis semakin tinggi, dan pot width akan tinggi. Di sini sudah menunjukkan tanda-tanda korelasi di sini. Nah, yang lebih menarik lagi adalah tentang chlorophyll retention. Bagaimana daun itu tetap hijau. Kami pilih 3 yang memang mewakili dari tahan kering, varietas tahan kering, medium, dan susceptible. Yaitu US 65 ini yang tahan kering, yang Lamongan ini yang medium, dan yang bulat kosong atau bulat putih adalah yang susceptible. Atau PE 337-409. Jadi yang segitiga hitam adalah US 605, itu drug toleran. Dilihat di sini bahwa retention klorofilnya itu lebih tinggi ya dibandingkan dengan itu. Jadi awal memang dia tinggi, tadi sesuai. Jadi laju penurunannya dia itu juga tidak sedrastis yang susceptible. Nah, tadi jatuh pada saat 7 hari. Dia sudah klorofilnya sudah sangat rendah. Itulah retentionnya sangat menarik sekali. Jadi agak lama penurunannya pada drug toleran yaitu US 605. Di sini yang itu yang tentang chlorophyll retention. Nah, sekarang bagaimana kandungan endogenous asam asiksit atau ABA hormon. Jadi ini kami ambil 2, yaitu bagian daun dan bagian akar. Sekali lagi, ini yang mewakili tahan kering, mewakili medium, tahan kering, dan mewakili yang susceptible pada kondisi kering. Jadi yang daun adalah yang arsir transparan, yang di arsir ini adalah yang akar. Di sini terlihat bahwa US yang tadi tahan kering, penelitian batu tahan kering, dia bisa memproduksi hormon ABA yang tinggi di daun. Ya. Jadi inilah dia itu. Dan hampir sedikit di akar. Itu juga Lamongan. Ini tengah-tengah dia ya, dia hanya mampus ini. Memang betul bahwa di daun itu adalah tempat fotosintesis, kelihatan, dan ABA adalah mengatur buka tutupnya stomata sehingga dia laju transpirasi bisa dikendalikan atau diminimumkan. Itu. Jadi itulah yang menarik dari ABA ini. Sehingga yang di drug sustainable ini jadi daun yang kecil kandungannya dengan akar yang besar di sini ya. Jadi tidak mampu mengkomunikasikan bahwa kondisi kering itu terpapar pada dirinya, yaitu susceptible one, yang susceptible kering. Sehingga transpirasi atau pembukaan stomata menjadi besar dan misalnya tinggi. Itu yang terpapar di sini yang menarik sekali. Inilah dia dengan melihat adanya kandungan endogenis ABA. Terus untuk hormon lainnya yang lebih dekat dengan kekeringan adalah cytokinin. Nah, sini. Cytokinin ini adalah di daun dan di akar. Jadi yang tahan kering itu sitokininnya adalah tinggi. Akar juga tinggi. Yang tidak tahan kering atau stable, sitokininnya rendah di daun. Di akarnya juga rendah. Jadi komunikasi lagi dua hormon itu adalah sitokinin dan ABA adalah lebih bagus di tahan kering. Ya, sitokinin dekat dengan kondisi klorofil, kehijauan daun ya. Jadi bagaimana mengatur daun agar tetap hijau, sitokinin dia dan tetap tumbuh. Sedangkan di ABA adalah ABA itu adalah mengatur stomata buka tutupnya, apertur. Itu yang di sini ya. Jadi dua hormon itulah yang mendukung tentang adanya drop tahan kering ya, drop toleran pada suatu varietas. Yaitu yang kita gunakan adalah kacang tanah. Dan itu didukung oleh jagung juga ABA, hormon ABA, di tempat sorghum juga hormon ABA, dan secara umum memang mendukung bahwa perannya adalah untuk buka tutup stomata sehingga dia mampu mengatasi transpirasi dalam kondisi kering, jadi dia akan misalnya akan sedikit ya dibandingkan yang tidak tahan kering. Jadi dua hormon itu yang dominan di sini. Itulah penelitian bagaimana laju fotosintesis bergerak untuk mempengaruhi hihin ya. Jadi lewat berbagai jalur sehingga itu mendeteksi bagaimana tanaman itu bisa tahan kering dan tanaman yang tidak tahan kering. Itu dia tentang kacang tanah ya. Nah, sekarang bagaimana tentang efisiensi penggunaan air dari 9 varietas tadi yang dimasukkan dalam 3 grup ya. Di sini bahwa penggunaan efisiensi air itu water use efisiensi, yaitu adalah rasio antara fotosintesis dan transpirasi itu lebih tinggi di drug toleran. Jadi memang mendukung ya di sini ya. Jadi dibandingkan dengan yang drug stable yang tidak tahan. Jadi water use efisiensi sangat rendah dibandingkan dengan drug toleran. Yaitu dalam kondisi optimum ini yang bersama ya, apalagi yang kubah dengan yang tahan itu sama dalam kondisi begitu ada kondisi kering maka dia akan berubah tergantung tadi ya dua hormon ABA dan sitokinin. Begitu dia sitokinin menjaga akar hijau, retention klorofil, IPA menjaga mengontrol buka tutup stomata sehingga laju fotosintesis akan membantu tanaman untuk tahan kering. Sebaliknya pada tanaman yang tidak tahan kering karena memang tidak ada komunikasi hormon ABA tentang buka tutupnya stomata, akibatnya begitu juga tentang sitokinin klorofil, maka dia akan menurunkan atau merendahkan seluruh komponen-komponen fotosintesis, ya. Termasuk laju fotosintesis itu sendiri. Jadi kelihatan di sini ya. Jadi dua hormon itu bekerja bersama untuk menciptakan kondisi tahan kering melalui perbaikan fotosintesis. Jadi dengan adanya mengukur fotosintesis, maka biar kita bisa cepat mendapatkan varietas yang produksi tinggi, terpapar di sini, tahan kering, ya. Nah, itulah adanya stomata, pendugaan Maka kami bertiga ini mengamati, yaitu Adi Saputra ini dari Universitas Negeri Jakarta, Pak Sudarsono Prof ini dari IPB biomolekuler, dan saya sendiri mengamati bagaimana pewarisan sifat stomata dan kelan air pada pinat. Nah, di sini jadi menyambung ya, jadi bagaimana tadi komponen fotosintesis yang tidak lain adalah stomata tadi itu bisa diturunkan nggak kira-kira kalau kita menyilangkan ke turunannya berikutnya. Jadi ini adalah cari di Indonesia yang memang tidak tahan pada kering, yaitu kelinci, susceptible. Dan yang tadi ini sebagai female ya, yang P1. Dan yang tadi US 605 yang bagus penampakannya di drug toleran sebagai sumber jantan. P1 betina, P2 jantan. Nah, inilah materi bagaimana F1 yang betina adalah kelinci, yang resiprok adalah yang betina yang US 605. Terus bagaimana di selfing, bagaimana di precross. Jadi kita amati dari materi-materi tersebut. Nah, penghitungan stomata atau densitas stomata kepada stomata adalah dengan imprint teknik. Ya, itu biasa ya. Jadi pakai kutek ya, dilekatkan pada permukaan daun. Nah, terus nanti itu disentek kuteknya dan dihitung. Dan laju kehilangan air dari daun, jadi dengan metode bagaimana daun direndam. Ya. Ini salah tulisannya, ini A nih, ini di dalam 30 meter HPS buffer. Ya. Terus diinkubasikan, lalu dihitung bobot daun awal, kemudian direndam pada 40% PEG, polietanol PEG, dan ada yang di ini sebagai PW. Jadi EW adalah bobot daun awal dan bobot daun setelah direndam PEG. Inilah laju kehilangan air dengan menggunakan rumus bagaimana pengurangan EW dengan PW dibagi dengan EW kali 100%. Itu yang rumus kehilangan air. Nah, ini terlihat bahwa pada kondisi stres ini ya, ini stomata density, ya ini ya, ini bahwa kalau persilangan P1 sebagai betina dan P2 sebagai male, jadi itu adalah densitasnya adalah ini ya 157 plus minus standar deviasi. Laju kehilangan air 22 ini. Dan kalau itu di resiprok, bagaimana persilangan? Hampir sama ya di sini. Jadi tidak ada berubah. Ini juga tidak ada berubah. Sehingga kalau dianalisis dengan T, ya, dihitung, ya tidak nyata. Tapi ya, tapi dengan adanya perbandingan tetua, maka dia nyata. Nah, memang, jadi tetua satu memang dia kelinci, itu adalah tidak tahan kering, dan B2 adalah tahan kering. Itu beda nyata. Sekarang persilangan. Nah, ini bagaimana P1 sini, maka P2 ini kecil ya. Maka F1 adalah dia berhasil menurunkan stomata dibandingkan dengan P1. Tapi bisa naik dibandingkan P2. Sehingga inilah dia derajat pewarisannya adalah minus. Ini ada semacam pengaruh pengaruh resesif. Jadi backcross-nya juga gitu. Backcross kalau dengan P1 maka dia akan lebih tinggi dibandingkan backcross dengan P2. Sehingga P2 itu kondisinya adalah terjadi segregasi. Sama dengan keadaan air ya. Inilah bentuk-bentuk stomata, bagaimana P1 yang lebih banyak stomata dibandingkan yang P2 US. Yang juga resiprok hampir sama, hampir mirip di sini kondisi stomata. Ini juga ini lebih ke arah yang P2, bagaimana P-cross ya, yang P2. Terus ini terjadi agregasi di F2 generasi, adalah stomata. Jadi di sini kelihatan bahwa stomata itu penting ya, penting, sehingga yang riset next adalah bagaimana besar atau apa ya, buka endoteliumnya semata itu bisa diukur dengan SEM, scanning elektron mikroskop. Nah, ini kami sudah nyoba ya. Jadi ini scanning mikroskop ini sudah nyoba kami mengukur tentang granul pada singkong. Jadi kalau itu memang pengukuran granul berhasil, maka next riset adalah bagaimana apakah memang varietas yang tahan kering itu suatu tanaman mempunyai stomata banyak tapi kecil-kecil, atau memang mempunyai stomata sedikit tapi besar-besar. Nah, itu dihitunginlah pengalaman dari Lampung mengukur SEM. Jadi nanti SEM pada granul. Jadi ini bukan ini hanya menunjukkan bahwa next riset itu adalah bagaimana buka besarnya stomata sendiri. Ini dia ya. Jadi berhasil bagaimana tipe granul di ubi kayu, tepung ubi kayu, start ya. Jadi pada kondisi 0 mikronutrien dia lebih kecil granulnya dibandingkan dengan 20 dosis 20 kg per hektar dan 40 kg per hektar. Jadi ini hanya menunjukkan bahwa potensi pengukuran stomata yang buka itu, besarnya stomata itu berapa itu ya. Jadi ke arah itu penting sekali. Apakah memang yang tahan kering itu sekali lagi banyak kecil-kecil mukanya atau memang sedikit besar mukanya stomata. Walaupun itu bisa diukur dengan Li-Cor ya. Jadi ini adalah fisik, bentuk fisik. Nah, yang terakhir yang sedang kami kerjakan adalah pengaruh langsung fotosintesis dengan komponen melalui PET analisis. Jadi ini masih. Jadi ada 4 genotip sorghum yang terpilih ini ya, genotip yang terpilih dari 35 sorghum. Jadi berdasarkan vegetatif, pertumbuhan vegetatif dan pertumbuhan generatif atau hasil. Terpilihlah jadi genotip harapan G7, GH13, B90 dan Super 2.
Prof.
Jadi pengukuran fotosintesis dengan leaf area index dengan 2200 plan canopy analyzer ini dari Li-Cor PCA.
Prof.
Dan fotosintesis rate dengan Li-Cor LI 6800 portable photosynthesis system. Jadi ini adalah percobaan lapang. Jadi inilah penggunaan sangat simpel ya dan nyaman. Bagaimana mengukur dengan Li-Cor yang ada di belakang saya, ini alatnya dan samping saya. Ini dia. Begitu juga bagaimana dia mengukur di daun bendera dan di bawah daun bendera. Yaitu untuk LAI bagaimana cahaya di atas permukaan daun dan di bawah tajuk itu ya LAI. Nah, ini kelihatan sekali bahwa bagaimana kuadrat tengah genotipe dari analisis ragam menunjukkan variasi. Jadi tinggi tanam ada variasi, tinggi tanam ada variasi ya, ada variasi leaf dieback juga ada variasi. Terus leaf area index tidak ada variasi. Ya, ini ya, tidak ada variasi di leaf area index. Kemungkinan ini memang satu dia diseleksi yang 4 unggul tadi ya dari 35 varietas. Jadi hampir sama semua di sini sorghum. Dan ini yang PET analisis dari kenyataan bahwa seed weight ini seed weight SW ini ya. Ini jadi langsung dipengaruhi oleh bobot dombolan, bobot head itu langsung dia. Dan leaf area index itu pengaruhnya melalui akar, bobot akar, baru ke bobot head dan ini. Jadi nggak langsung dia leaf area index. Begitu juga kita bagaimana fotosintesis. Jadi fotosintesis ini adalah ada variasi dia. Begitu juga ada transminasi, ada variasinya juga. Itulah dia komponen fotosintesis yang diukur dengan Li-Cor 6800. Nah, di sini bagaimana di daun bendera, ya, ini flag, anti flag leaf, dan ini adalah di bawah daun bendera. Jadi ini adalah fotosintesis ya, di sini. Dan ini adalah transminasi. Kelihatan bahwa super 2 ini walaupun fotosintesisnya rendah, tapi transminasinya juga rendah di sini. Super 2 dibandingkan dengan yang 2, GH13 dan GH7. Di sini kenyataan bahwa terlihat bahwa water use efficiency super 2 lah yang lebih tinggi. Jadi di antara seleksi 35 untuk unggul tadi ya, yang 4 terpilih, rupanya super 2 yang paling unggul di sini berdasarkan fotosintesis dan transminasi rasio. Itu yang menarik sekali di sini.
Maaf Profesor, waktunya udah ini Pak, hampir habis Pak.
Yo, ini dari PET analisis terlihat bahwa biji tidak langsung dimulai oleh fotosintesis, tapi dia melalui jumlah biji dan stem diameter, jadi nggak langsung ya dari fotosintesis. Jadi itu kesimpulannya bahwa memang dari penggunaan alat ya, semakin lama waktu ya, tadi semakin nyaman ya bagaimana cara mengukur fotosintesis. Jadi fotosintesis daun komponennya itu berkorelasi dengan growth dan komponen hasil, dan bisa dipakai sebagai kriteria high yielding variety atau drop di sini ya. Dan salah satu komponen utama fotosintesis adalah stomata yang tadi diwariskan secara genetik. Dan fotosintesis komponen menunjukkan pengaruh yang tidak langsung kepada hasil melalui PET analisis. Jadi ini adalah gambaran kami juga mengukur penelitian fotosintesis untuk cassava beksi dan non beksi. Nah, ini cara mengukur LAI, yang mengukur dia di bawah tajuk dan di atas tajuk, terus dia mengukur fotosintesis leaf dengan Li-Cor 6800. Ini juga dia nyaman, jadi tinggal dasinya. Dan ini penggunaan elit, ya, pengukuran alat-alatnya, jadi dua kali pengukuran, 4 minggu dan 10 minggu tanam. Ini hasilnya belum ada, tapi kelihatan menjanjikan dalam waktu dekat akan terpapar nanti, dipublikasikan. Dan terima kasih saya nyatakan kepada Rektor Universitas Lampung yang untuk kesempatan bagus ini. Kedua adalah Dekan Fakultas Pertanian Universitas Lampung yang mensupport webinar ini, dan Kepala UPT Laboratorium Terpadu dan Sentra Inovasi Teknologi yang menyediakan fasilitas dan sebagainya. Dan terutama juga terima kasih pada Unitama yang bekerja sama, dan teman-teman saya Profesor Wilasono dan Dr. Adi Saputra untuk kerjasama riset. Ini juga teman saya yang mendukung adalah Profesor Kamal dan Dr. Samsuladi yang untuk kerjasama sini. Begitu juga murid saya yang dari kerjasama, Restu, Amira, Aji, dan Indah untuk mereka juga. Jadi itulah sementara yang bisa saya sharing dan mudah-mudahan bermanfaat. Saya mengucapkan terima kasih dan akhir kata, Assalamualaikum warahmatullahi wabarakatuh.
Baiklah, Bapak-bapak dan Ibu-ibu sekalian, dan kepada Profesor Kukuh Setiawan kami mengucapkan terima kasih atas presentasinya yang cukup menarik, dan tentu saja mungkin ada beberapa peserta yang akan bertanya, tetapi mungkin karena masalah waktu kita pertanyaannya nanti kita sampaikan mungkin pada saat sesi question dan answer, ya sesudah nanti presentasi dari Li-Cor ini. Baiklah, untuk menyingkat waktu kita lanjutkan dengan sesi berikutnya nanti dari Li-Cor. Oke, Jason, you are ready? Jason.
Good morning.
Morning. Yeah. Now is now your session for your presentation. Yes. The time is maybe for until 11:40 p.m. Yeah, for you. Okay. Okay. Time is yours.
Can we see my presentation?
Yes. Yes. That's right.
Very good.
Okay.
So, hello, everybody. I'm Jason. I'm a scientist at Li-Cor. I was involved in the development of two of the instruments that we're going to talk about. And yeah, so we'll just jump right in. So I've sort of divided topics. Today, we're going to introduce sort of the operating principles and theory of some of the key measurements we make. And then tomorrow we'll talk about sort of the environment and the different kind of data that we can get from the instruments. And oops. I'll just start by saying that I'm a botanist by training, so I love math. That's what they teach you in botany school. And I have lots of equations in my presentation. I've tried to make these as clear as I can. I like them because they're the best way to illustrate many of these topics. A number of them are presented in their simplest form, and the idea is that we're using these for troubleshooting and data quality tools. And the way that I present on these topics is generally structured around what I think of as sort of the errors in these measurements. You know, when I approach something, I generally think about it.
Sekarang iya bisa nih sekarang. Setelah saya restart.
From the context of what could go wrong before I assume what I'm doing is right. And so when I think about leaf level physiological measurements, the things that we're going to talk about, I sort of split the errors out into three categories. There's sort of what we call instrumentation errors. These are things that relate to a problem with the instrument, maybe an issue with its calibration, not understanding that something's a computed parameter versus something that's directly measured, and the underlying assumptions that go with that. And then I think about things like interpretation errors, and this is where I'm getting data from the instrument, and there's some fundamental assumption that goes into getting that data that I've just missed. And then I think about application errors, and these really come down to how we apply the instrument to make a particular measurement. And so as we go through, I'm going to talk about a whole bunch of things that relate to these individual sort of error categories. The things for today will mostly be instrumentation or interpretation related, and then there'll be lots of discussion of application tomorrow. So you've already gotten a little taste of this, but how do we measure photochemistry? We take, with Li-Cor instruments, we take two general approaches to this. So one of those approaches is to measure this thing that we call gas exchange. And in the case of a Li-Cor instrument, we're measuring the exchange of water vapor between the leaf and the atmosphere, or the exchange of carbon dioxide between the leaf and the atmosphere. And this gives us information about the diffusive pathway between the atmosphere and the leaf. It gives us information about the state of carboxylation. And then the other thing that we measure is called chlorophyll fluorescence. And this is a technique that allows us to probe the state of photosystem 2 directly, and it gives us information about the efficiency of energy capture and conversion to do photochemical work. And from that efficiency, we can start to derive downstream processes from that. And I'll go into fairly great detail about what I mean. I'll start by saying that we currently make 2 instruments at Li-Cor that are combined gas exchange and fluorescence instruments. One of these is the Li-6800 portable photosynthesis system, which is sitting behind me here, clamped on some lemongrass, which surprisingly for 10 o'clock at night in Nebraska has a photosynthetic rate of about 11 right now. And the other instrument we make is the Li-600 porometer fluorometer. And we'll talk about that instrument in detail tomorrow, and I'll show you some example data sets from it. So I'll start with chlorophyll fluorescence. And anytime I talk about this, I like to start with a pretty picture, because I find chlorophyll fluorescence to be a terribly abstract topic. It took me many years to really wrap my head around what was happening here and how it worked, and I'm still not convinced that I fully understand it. And, you know, most people, we see the world and we see green plants everywhere, and we're completely unaware that if you have the right kind of eyes, that plants are pink, right? If we illuminate chlorophyll, it starts to emit photons through this process that we call chlorophyll fluorescence. And you can see it here. So this is a beaker of spinach chloroplast extracted in acetone. And on one side, it's just photographed under white light. That's the way that we would normally see it. In the other image, there's a long pass filter. So it's cutting off all light below 700 nanometers wavelength. And we see that the beaker goes from being green to pink. And it's pink because that chlorophyll is re-emitting photons as a means of getting rid of that energy. If we think about sort of an individual chlorophyll molecule, we can make this stylized diagram of its various energy states. And S0 would be the ground state. And you can see I've got a nice arrow here representing absorption of a blue photon and one representing absorption of a red photon, right? So blue light is relatively short wavelength, high energy. Red light being longer wavelength, lower energy, but they both have enough energy to get chlorophyll excited to the first singlet state. And once it's excited to that first singlet state, then that's when it can donate an electron eventually to an open reaction center and do what we typically think of, right? So it's captured that photon, that's excited it, now it's going to release an electron to the electron transport chain, and it's going to do photochemistry with it. But that's not the only pathway of energy dissipation. So I have a whole bunch of pathways here that are drawn as green arrows that I call non-radiative decay. These are a number of different processes, but ultimately they all end up as heat being dissipated from the leaf. And then there's an arrow drawn in sort of dark red that's labeled fluorescence. And that's re-emission of a photon from that first singlet state to get back to the ground state. So all of these pathways exist in parallel. And whoever gets the energy from a given absorption event by a given chlorophyll molecule is whoever's fastest at that moment in time. So when we work with chlorophyll fluorescence, we typically think about it in the context of rate constants. And here I have 3 different equations that basically say the same thing in the end, and that is that some yield of fluorescence, so those photons coming back off the leaf, is a function of all the various energy dissipation pathways in operation at the given point in time. And here we have a rate constant defined for dissipation via fluorescence. We have a rate constant defined for intersystem or for internal conversion and intersystem crossing. Collectively, those are those green arrows that we show as heat dissipation. We have an apparent rate constant for chemistry. That's the thing we're generally interested in. And then we have an apparent rate constant for non-photochemical quenching. So this is energy dissipation through a number of inducible pathways that also ultimately end up as heat, but they get used differently than these guys do. So these form, this equation here forms our governing principle of chlorophyll fluorescence. And we call the photochemistry and non-photochemical rate constants apparent. That's why they're denoted with a prime, because they're not constant. You can think of each one being multiplied by a scaling factor that has to do with the degree of engagement of that pathway. This becomes a really important concept as we get to defining the things that we measure from chlorophyll fluorescence. We'll get there in a second. So let's talk a little bit about how a fluorometer works. And you can see on my instrument setting here, so this is the chlorophyll fluorometer. This is what we call a PAM fluorometer. It uses what's called the pulse amplitude modulation fluorometry technique. And that's just a fancy way of describing the different kinds of light that get applied in the measurement. So a fluorometer is not a particularly complex device. In our case, it's an array of LEDs, so light emitting diodes. We typically have a lot of red ones, a few blue ones, and then one or two far red ones. And these are at like 730, 735 nanometers wavelength. These shine light down onto the leaf. That induces the leaf to do a number of things. One of them is emit fluorescent photons. Some of those photons go up. They pass through, in our case, a long pass filter. So this blocks out all light below 700 nanometers. And then we capture what comes through a detector, right? Somebody's drawing on my slide, okay. So we apply these red lights in two different ways. We have what we call the DC component to the light and the AC component to the light. So the AC component is where the modulated comes from in the PAM fluorescence, right? Pulse amplitude modulation. So this is light that's turning on and off very fast. So typically this light is turning on and off somewhere between 50 and 5,000 times a second, such that it has a mean light intensity that's very, very dim. It's just enough light to cause a little rise in the fluorescence. You can see down here, there's this little wiggle that it induces in the fluorescence, but it's very little. So the mean intensities here are on the order of maybe 0.5 ppfd. If full sunlight's 2000 ppfd, that's pretty much in the dark. And then the pulse part of this PAM comes from these DC lights. And this is where we turn the red lights on very bright for a very short period of time, typically less than a second. And it is a very high intensity. So this is somewhere between maybe 8 and 16,000 ppfd. So 4 to 8 times full sun intensity. And that causes a dramatic rise in the fluorescence. When we apply this pulse to a leaf that's been setting in the dark, we would call this a dark adapted measurement. And we can make an analogous measurement in the light. The difference being that some portion of these red LEDs is on somewhere between 0 and full sun intensity. And then we apply the pulse on top of that. In real time, the instrument's giving us a trace of fluorescence that looks something like this. It's not very interpretable. What we use is this little wiggle to pick off the fluorescence we're inducing. So we get discrete points in time. And this bottom panel is where all of our raw fluorescence measurements come from. From this bottom panel, we derive these parameters that you might see in the data file if you've looked at this. We get from that modulated fluorescence in the dark, remember just that little tiny bit of light, maybe half a ppfd. We get a parameter we call F0, and this is minimum fluorescence in the dark. When we apply the pulse, the fluorescence rises to a maximum value that we call FM. The difference between F0 and FM is the variable fluorescence called FV. In the light, from that modulation, we get a parameter we call FS, steady state fluorescence in the light. When we apply a saturating pulse, we get a parameter that we call FM prime, the maximum fluorescence in the light. The difference between those is delta F. And then from a special measurement that I won't go into detail here, we get a parameter we call F0 prime, which is minimum fluorescence in the light. So this seems like maybe I'm jumping ahead in getting here, but I want to tie this around to the physiology. So each one of these parameters, F0, FM, FS, FM prime, they represent our leaf in a discrete physiological state. And we've used light and tricks of light to drive the leaf into that state. Each one of these represents a unique balance of those rate constants, that basic formulation I showed you. Right? And the difference between these parameters tells us information about the capacity of photochemistry inside our leaf. So let's look back at our rate constants. So here are our 4 basic fluorescence parameters, F0, FM, FS, FM prime, right? So these are dark adapted, measured on a leaf that's been in the dark. These are light adapted, measured under sort of ambient operating conditions, right? And you can see F0, FM prime, well, what's the difference here? This thing called KPC, the rate constant for photochemistry, has disappeared, right? So when we apply that little wiggle of light in the dark, you'll note that there's no PQA here. The assumption of measuring F0 is that we're measuring the leaf in a fully relaxed state. We mean that every reaction center is open and waiting to accept an electron to do photochemical work, such that the rate constant for photochemistry is at its maximum value. Then we apply that saturating pulse, that really high intensity light. I said 4 to 8 times full sun intensity. The assumption there is that the light is so intense that this multiplier on KPC goes to 0. We filled all the open reaction centers instantaneously with electrons. And when we do that, we've killed the leaf's capacity to do photochemistry for that moment in time, and it drops out of the equation. Now, what this tells us is that the difference between F0 and FM is a measure of capacity to do photochemistry. Similar measurement in the light, and you'll note that the difference here is we include this NPQ parameter, right? Non-photochemical quenching is a collection of inducible processes that plants do in the light. Okay? So these equations highlight the underlying assumptions of the measurements that we make, right? To measure F0, we assume that PQA equals 1. When we measure FM or FM prime, we've made the assumption that PQA is driven to 0, such that when we multiply it by KPC, 2 equals 0. When you go to make measurements with a fluorometer, these are probably the first 2 assumptions that you should test. And you can do it with the instrument. It's really cool. This is data where I took a low light adapted plant. It's a little tropical gesneriad in the genus Apishka, and a high light adapted plant, soybean. And I put a dark adapted leaf in the chamber. So that just means I kept it in the dark for about an hour. And then I turned the modulated light on the instrument, that little wiggle. And I increased the modulation frequency, which in turn increases the amount of light that I'm giving the leaf. Here, this is about 0.05 ppfd. No, 0.005 ppfd. And this is 0.5 ppfd, right? It's still dark. And I let it set at each little modulation frequency for a few minutes, and then I up it. And what we should see, if we're not inducing photochemistry, right? We're not violating the pool of open reaction centers equals 1, is that as we increase the light, there should be a little rise in fluorescence, and then it should come to a steady state value. And the rise happens because we put more photons in, so we expect to get more photons back out. But you can see in this low light adapted plant, this Apishka, when I go from 500 to 1 kilohertz, 500 to 1,000 hertz, fluorescence rises, and then it starts to decline. And then to do it again, and it declines more. What you're seeing here is that light energy that's being added to the leaf is now being dissipated through photochemistry. We're actually providing enough light at 0.1 ppfd to make this particular leaf do photosynthesis with it. Now our soybean, it's used to a lot more light. And you can see as we step it up, it doesn't do anything. We just get a higher fluorescence yield. It's not enough light for it to actually do photochemistry. If I was going to measure this soybean, I would probably use a modulation rate of 2 or 5,000 hertz, somewhere in there. If I were going to measure this Apishka, I certainly wouldn't use anything above 500 because I know it induces photochemistry. When we test for the assumption that our flash is saturating, that we're driving photochemistry to 0 when we provide the pulse of light, we typically just iterate over the flash intensity. This is something you as a user can set up. You can script it with an auto program. And how to do it. And we'll just let measure the as a flash intensity. And you can see I'm using the same plants here, my low light adapted Apishka and my high light adapted soybean. And as we might expect for a low light adapted plant, it doesn't matter what flash intensity I get. I always get the same FM prime. But the soybean does something different, right? There's some kinetics here. As I start to increase my flash intensity, FM prime. Long ago, so long ago, Joe Barry recognized this phenomenon and developed a technique called the flash train. And basically, he did this experiment right here. But instead of looking at it at the flash intensity, he looked at FM prime over 1 over the flash intensity, the inverse. So now here's my lowest light point, and here's my highest light point. This linearizes the curve. And if I extrapolate it out to the y-axis, it tells me what FM prime would be if I could provide an infinite amount of photons to the leaf, right? So we can see in the soybean example here, there's not a whole lot of difference between what I get at this high flash intensity and what that intercept is. As I move to something like a C4 plant, like sorghum or corn that's field grown, what you'll generally see is that the slope of this line will get steeper. And at the saturating intensity of the fluorometer, you might not actually see a really tight agreement between these. On our instruments, we support a technique to deal with this called the multi-phase flash technique. And instead of doing a flash train, like my previous experiment that takes an hour or so to get 1 point, this does the flash train by modulating the light intensity in the time span of a normal rectangular flash, a normal saturating pulse. So less than a second, we do this linear ramp in intensity, and we fit it to get FM prime at infinite light intensity. And so if you're working with field-grown C4 plants, this is something that you probably want to look at after doing the experiment from the previous slide. So why do we measure chlorophyll fluorescence? Well, we use it for a lot of things. It can be a particularly sensitive indicator of stress. And it can tell us a lot about how the plant is dealing with stress. It can also give us information about how energy capture is coupled to carbon capture. And so I'll just start here. This is probably the most widely cited parameter from chlorophyll fluorescence, this thing called FV over FM. It's the maximum quantum efficiency. Measured under a completely relaxed state, it tells you the maximum potential your plant has for capturing light energy and converting it to do photochemical work. And this is the most widely cited parameter because evolutionarily, it's incredibly highly conserved. If you go measure healthy plant material pretty much anywhere on Earth, regardless of taxa or location, FV over FM will be about 0.8, suggesting that intrinsically plants are about 80% efficient at capturing light to do photochemistry. Converting light to do photochemistry, I should say. So FV over FM is in a fully relaxed state. Its counterpart in the light, when the leaf is doing photosynthesis, is called phi PS2. That's the operating efficiency. And phi PS2 is inversely related to light intensity. As you start to give the plant more light, it becomes less efficient at using it. I'll show some data tomorrow from a light response curve, and we'll look at carbon assimilation and phi PS2 and see how they operate inversely to each other. From phi PS2, if we know something about how photons are partitioned between PS1 and PS2 and how our leaf absorbs light, we can derive a parameter called electron transport rate. And this is a count of electrons moving down the electron transport chain. This parameter is neat for a whole number of reasons. One of them is that based on photosynthetic metabolism, there's a theoretical minimum electron cost per carbon gain. I have some data on the screen here, measurement tobacco. And I made these measurements under low oxygen. That's a key point here. 0.5% oxygen, not 21%. And you can see in the bottom panel, plotted electron transport rate versus gross assimilation. We can see that the slope of that line is 4. Well, the theoretical minimum for a C3 plant is 4 electrons per carbon fixed. To get 4 electrons down the electron transport chain, you have to capture 8 photons. Look at that top plot. It says 8 photons per carbon fixed, right? So this plot was done under what we would call non-photorespiratory conditions, meaning that I deprived the leaf of oxygen so that it couldn't do photorespiration. If we make the same measurement at ambient oxygen, we see the slope of this line increase. The steepness of this line relative to the theoretical minimum tells us something about alternative processes sinking electrons. In a C3 plant, that's predominantly photorespiration. When we make measurements in the light, in the dark, and combine them, we can derive parameters like NPQ, this metric called non-photochemical quenching. Collectively, this is a whole bunch of stuff that dissipates energy ultimately as heat. But plants use NPQ to do a number of things. It's a conglomerate of a number of physiological processes. One of them that they use it for is to balance the energetic outputs of photochemistry, meaning that part of NPQ is related to photorespiration, and they actively engage and disengage NPQ to balance the ratio of ATP versus NADPH produced. That's pretty cool. As stress increases, what we see is a concurrent increase in NPQ. We can get parameters like QL or 1 minus QL. This tells us the fraction of open or closed reaction centers. This gives us information about, we make these as relative measurements between different leaves. Under stress conditions, it gives us information about demand for energy. Some points of caution that I like to point out with fluorescence. is that unlike all the other instrumentation we'll talk about, fluorometers aren't calibrated to any standard. That's why all those parameters on the previous slide were functionally ratios, right? We measure the leaf at one state, we count some photons. We measure a leaf at another state, we get a photon count. We take the ratio of those two. They're not scaled to engineering units. This means for things like NPQ, if you want to measure them, and you're going to go make a measurement in the dark and a measurement in the light, but we generally want to do that with the same fluorometer configured in the same way. Otherwise, we add an error to the measurement. For a number of things on our instruments, we include default values, like that alpha value in the ETR calculation. We include default values. They're different depending on what instrument, what light source is being used. But they're defaults. And I would encourage you to look outside and decide whether or not all leaves are ultimately the same shade of green. If you think they're all the same shade of green, then you're probably okay using our default absorptance. If you don't think they're the same shade of green, then I would encourage you to measure absorptance if you want to characterize something like ETR. And the tool used to do that is an integrating sphere and a spectroradiometer. Some of the fluorescence parameters, like phi CO2, require an estimate of dark respiration. We include a default value of 1, but this is simple enough to measure because you just turn the light off and you measure the carbon assimilation in the dark. And that gives you a pretty good estimate of dark respiration. If you're starting out in fluorescence measurements, there's a few handy references that I like to point people to. So the first two are reviews of the PAM chlorophyll fluorescent technique. One is an updated version of the other, but I think they're still both useful to go and review. The third one, this Perkasa paper, is actually a SIF paper. It's about solar-induced fluorescence, but they provide a really nice discussion of this rate constant, defining the traditional fluorescence parameters. So I like the introduction to that as a good start resource. And then this Baker paper is a really nice review of individual chlorophyll fluorescence measurements and how they've been used to examine stress in various crop applications. And so they go through everything like FP over FM and phi PS2 and so on and so forth. And they cite particular examples and how it ties to the particular stress that the researcher was looking at. It's a really great resource too. So we'll shift gears now. My equations change color. New topic. And we'll talk about measuring gas exchange. So we'll spend a lot of time talking about gas exchange because fluorescence is pretty straightforward. You know, you mind some assumptions and you hit a few buttons and you get reasonable numbers out of it, right? The application of the instrument to do it is straightforward. The underlying theory is incredibly dense. Gas exchange, on the other hand, the underlying theory is pretty straightforward. But there's a lot of parts to make the measurement that interact with each other. And then there's a lot of opportunity for how we apply those parts to the leaf to change the outcome. And so I generally like to say that gas exchange is sort of fine art. I've been doing gas exchange measurements for 20 years now, and I still learn new things all the time. So maybe some of you have seen this equation before. If you've taken a chemistry class at any point, you've probably seen some formulation of it. This is just Beer-Lambert's law. And it says that the absorption of light at some wavelength or range of wavelengths is a function of an absorption coefficient at those wavelengths. The distance the light had to travel through the absorbing medium, and the density of the absorber. And I really like this equation, this formulation of this equation to start this discussion of gas exchange because it points out a really key feature of a gas exchange instrument. That while when we look at them, and we'll see data from them, and you saw data earlier, and those data were presented in molar units. right? We think about concentrations in mole fraction, ppm, micromoles per mole, micromoles of CO2 per mole of air, or millimoles of water per mole of air. But fundamentally, our instrument measures density, right? Density is like moles per cubic meter. And to get from this to the thing that it reports, we have to apply the ideal gas law. And I like to start here because I like to tell people that if you're going to go take one of these instruments to the field and make measurements with it, you should probably check to make sure that it's reporting a reasonable pressure and a reasonable temperature to start with. Because if it doesn't, there's a real issue. Because it's measuring density and it's applying the ideal gas law fundamentally to get to mole fraction. Everything that we do is going to be computed from mole fraction. So this Beer-Lambert's law works for our gas measurements because heteratomic molecules like carbon dioxide and water vapor absorb light in the infrared, right? So this is an absorption spectra for carbon dioxide in the bulk atmosphere. You can see what we would call absorption bands here, these sort of regions of things where light's being absorbed. If we zoom in on one of these bands, we see that it's composed of what at this scale look like a whole bunch of discrete lines. And indeed, we call these absorption lines. And you can see there's a number of them here, right? In various distributions, right? These different distributions represent different isotopologues of carbon dioxide. Each one of these lines represents a particular rotational or vibrational transition that the molecule goes through. And they're fundamentally mass-based, right? So if we have different isotopologues that have different masses to them, then that changes how the molecule rotates and vibrates, and it changes how it absorbs infrared light. If we zoom in on one of these lines, they're not discrete in nature, right? They have a Lorentzian or a Gaussian distribution to them. It's a tight bell-shaped curve. And it doesn't matter that you know this in detail, but sometimes I think it's good to present more detail than what we actually need. Because what I'm going to do is I'm going to come back to this alpha thing, right? We're going to talk about spectroscopic corrections for water vapor and oxygen. And we're going to come back to this alpha. But before we do that, I want to talk a little bit about how we go from this fundamental principle of Beer's law to an instrument that measures a gas, right? So we have to package the principle up into a device. And in the LI 6800, that packaged principle is called an infrared gas analyzer. And we use an optical differential technique there. That just means that we use optics to measure a difference. So a gas analyzer is a fairly simple device, fundamentally. There's an infrared source, the little red squiggle here, and that's just a heated filament. So you can think about it as an incandescent light bulb, but not a very good one. It produces a lot of heat and not a lot of visible light. And opposite of that infrared source is a pyrolytic detector. You can think about that as a solar panel for heat. So it captures the infrared light, the heat emitted by this filament, and it converts it to an electrical current. And the more heat that hits it, the more current that comes out of it. Okay? Between the source and the detector, there's a volume that we call the optical path, where we're flowing our gas that we want to measure through, right? That path is sealed by some optics at either end that keep this beam of heat, infrared light, narrow, tight, so that it goes through. And between the source and that optical path is a filter wheel or a chopper wheel. Here it is on the side, and here it is looking flat on. And basically it's a disc with 4 windows in it, and each one of those windows contains a piece of glass, an optical filter that lets through light above a wavelength and below a wavelength, so that we're looking at a band of light, and we're not seeing anything outside of it. And there's 4 of them because we measure 2 gases. We measure carbon dioxide, and we measure water vapor. And there's 2 windows for each gas. So one of those windows for carbon dioxide is in a region where it absorbs infrared light, but water vapor doesn't. And one of them for carbon dioxide is in a region where neither carbon dioxide nor water vapor absorb infrared light. So we call the one where they absorb the absorbing filter, and the one where they don't absorb the non-absorbing filter. We make the same pairing for water, and we get that absorptance value for Beer's law by looking at the amount of light that comes through when the absorbing filter is in place, and when the amount of light that comes through when the non-absorbing filter is in place. And 1 minus that ratio is absorptance, right? And we know that from Beer's law that absorptance is related to the gas density, and through the ideal gas law, that density is related to the mole fraction that we report on the instrument. So I like to talk about this because we're going to take these instruments out in very dirty places to make measurements. And there's some filters in them, but when we look at the flow path, you'll see that there's not much opportunity to filter the air between where the leaf is and where we make the measurement. We use this optical technique because it doesn't matter if a piece of dirt is setting in here, right? If I have a big chunk of dirt in here, well, it's going to block the same amount of light in my absorbing band as in my non-absorbing band, because a big chunk of dirt is spectrally neutral. And so I'll get the same absorptance. The measurement will get a little noisier because my signal goes down, but I get the same concentration in the end. This technique gives us field robustness. So to get from a gas analyzer to an instrument that measures gas exchange, we have to package it into a system. And historically, the oldest forms of gas exchange systems are based on what's called a closed transient technique. And this is like the Li 6200 that was referenced in the previous talk. In a 6200, we have a box, a cuvette or a chamber. This is just a clear box that we put a leaf in, and then we circulate air between that box and the gas analyzer, this sort of khaki colored piece here, and back to the box. And that air circulates in a closed loop such that if we put a leaf inside there and it is happy and healthy and in the light, it'll start to pull down the CO2 concentration in it, right? And the slope of this line is related to the rate of carbon assimilation, such that if we took the slope and we multiplied out by the ideal gas law and we divided by the surface area of the leaf, we get the assimilation rate. But you'll note that I didn't draw this line as a straight line, right? It has a curvature to it. That should imply that as we put the leaf in the box, the carbon assimilation rate, the photosynthetic rate's changing, right? Because there's a biological feedback between the carbon dioxide concentration outside the leaf and the rate of carbon assimilation that the leaf is doing, right? Some of this is purely diffusive. It relates to Fick's law. We'll talk about that in a moment. And some of it's biological. As you decrease the CO2 concentration, you start to down-regulate rubisco. And so people recognize this, and they took this basic architecture and they converted it to what's called a compensating system. So here, they bleed CO2 back into the instrument to keep the slope of this line 0. And whatever rate you have to add pure CO2, that's the rate at which the leaf is taking it out. That gives you the assimilation rate. There were a few commercial compensating systems, but none of them were particularly fieldable. They were big. Pretty much all commercial instruments now use what's called an open flow through or a steady state approach to the measurement. And that's what the 6800 uses. That's what the 6400 used before it, right? In an open system, we still have a box that we put the leaf in, our cuvette, but we have a constant flow of air into that box and out of that box, right? And that air enters with some carbon dioxide and some water vapor concentration. It interacts with the leaf while it's inside the box. If the leaf is happy and healthy and in the light, it will start to release water vapor through transpiration E, and it will take up carbon through assimilation A, such that as the air interacts with the leaf, it becomes depleted in carbon dioxide and enriched in water vapor. And when it gets forced out the other side, it now leaves at a different carbon dioxide and a different water vapor concentration. If we take the difference between what goes in and what goes out, and we divide by the surface area of the leaf, we can compute our flux. Now, in our instruments, you'll note that we really only report one flow rate. And that's because to measure this difference in flow rate with enough accuracy to actually be able to compute these things, the technology doesn't exist, right? So we apply some mathemagic, fancy mass balance, to calculate the fluxes accounting for the additional molecules leaving the system. Basically, we assume that the change in CO2 is so tiny it doesn't make a difference. It doesn't. It's generally like 10 to the minus fourth, 10 to the minus fifth. But the change in water vapor is pretty significant. It'll be somewhere on the order of 10 to the minus first or 10 to the minus second if you didn't account for it. A big enough error that we have to account for it. So we get these equations. These are, in SI units, the way these fluxes are calculated on the instrument. And I love these. I think that as a user of one of our instruments, this is maybe the most, one of the most valuable and fundamental things to sort of get ingrained into your head before you go out into the field. Because it highlights sort of maybe the key feature of the instrument that's really necessary to understand to ensure that you're getting quality data from it. And that is that the instrument doesn't measure an assimilation rate. It doesn't measure a transpiration rate. These are outputs. They're computed based on the difference in concentration of an airstream before and after it interacts with the leaf, right? These terms I have highlighted in blue we call deltas. Delta H2O or delta CO2, right? And they're the fundamental signal the instrument measures. It turns out that you as a user have a lot of control over how big these deltas are. That probably sounds counterintuitive the first time you hear it. It certainly did to me a long time ago. But you have a lot of control over how much leaf area goes in the chamber, and the flow rate is a completely user controllable parameter, right? And that means that for any given assimilation rate, if I put more leaf area in the chamber, this delta gets bigger. If I turn this flow rate up and make the flow rate larger, faster through the chamber, then this delta gets smaller. But the assimilation rate doesn't change. And so this is data from capsicum, bell pepper, where I just did a simple experiment. I put it at a happy light intensity, maybe 2/3 saturating. And I measured the carbon assimilation rate in green as I varied the flow rate through the chamber, right? Down here on this axis. And plotted in blue are the deltas, right? These guys, for CO2. And my assimilation rate's constant, right? There's no biological feedback to flow rate in the chamber. This is exactly what I'd expect, no change. But as I change the flow rate, the delta changes pretty dramatically. At my lowest flow rate, it's about 35. That's huge. At my highest flow rate, it's 10. That's still pretty good. But what's the trade-off? Well, I took the instrument noise specification and I just modeled some error bars on the assimilation rate. And if I assume a peak-to-peak noise of 0.3 ppm, this is 6400 data, 6800 is a little better. But I assume 0.3 ppm peak-to-peak noise. At this lowest delta, the highest flow rate, you know, there's some error bars. We can see them. It's about plus or minus a little over half an assimilation unit. As we go to this highest flow rate, the error bars become so tiny that we can't even see them behind the point that I plotted. So when we think about making these measurements, one of the key hurdles to get over as a new user is to think about assimilation as an output. It's not, I went to the field and I measured an assimilation of X, this is good or this is bad. It's I went to the field and I measured an assimilation rate of X, and I thought about how much leaf area I had in my chamber and what flow rate I was using to try and make this number as big as possible so that I know when I measured this assimilation rate, I did it as best I can. So I've rewritten the mass balance equation here to make it a little bit more instrument friendly. And so this is scaled to the units we use on the instrument. And I've changed the notation. In Li-Cor instruments, we call what goes into the chamber reference and what's in the chamber sample. And what's in the chamber should be representative of what's coming out of the chamber. And we use this reference and sample terminology because of the way we lay out the flow path in the instruments. And I'll talk about that in a couple of slides. And then before I get there, I want to come back to this point about this term in parentheses here. So this term for water vapor is used to adjust the sample CO2 concentration to the water vapor concentration in the reference. So fundamentally, we use a theoretical CO2 concentration here. We call this a dilution correction. And it has to do with the effect of water on the system. So if we're measuring a carbon assimilation rate and a transpiration rate, that transpiration rate is generally 10 to the third to 10 to the fourth times larger than our assimilation rate. Or another way to say that is that for every carbon that goes into our leaf, the leaf is releasing somewhere between 1,000 and 10,000 water vapor molecules, right? This makes the flow rate out of the chamber larger than the flow rate into the chamber. And because the chamber is well mixed, that volumetric dilution by water is forcing CO2 out the exhaust. And if we don't account for it, it appears as uptake by the leaf. And I talk about this because I've had more than one conversation over the years with somebody that didn't believe me about this. I don't know why you wouldn't believe it. The math should speak for itself. But just to hit on it, this is data from wet filter paper. Basically a paper towel that I got wet and I put in the chamber. And nobody ever argues that wet filter paper does photosynthesis. It doesn't take up appreciable amounts of carbon. Right? So the assimilation rate should be 0. And if you use the equation as we have it implemented on the instrument, you get a rate of essentially 0 within the limit of resolution for how it's set up. But if you use the basic mass balance, ignoring dilution, you'll see that the assimilation rate is directly proportional to the transpiration rate. Not a good thing, not real. This is volumetric dilution. And this is why we include this term in the mass balance. So I promised, for better or worse, that I would come back to this thing called alpha here and talk about these absorption lines. So the dilution correction isn't the only water vapor correction we make on the instrument. It turns out that because these absorption lines relate to physical motion of the atoms in the molecule as they move about their bonds, that they're influenced by collisions with other molecules in the gas mixture, such that if you change the amount of water vapor in a volume of gas, but you keep the density of CO2 the same, the amount of infrared light that CO2 will absorb changes as you change the water vapor concentration. And that's because as those water vapor molecules more frequently or less frequently collide with the CO2 as their concentration changes, it changes the shape of these lines, how wide they are, this alpha, the half width. That's great. We measure water vapor on the instrument and we correct for it in this process called band broadening, a band broadening correction. There's a really common measurement that people make. I showed data in my fluorescence discussion about non-photorespiratory conditions where I change the oxygen concentration. And I like to talk about this when I'm introducing the instrument to new users because it's important to tell the instrument how much oxygen's in the air when you go to make the measurement. The default value is 21% because that's atmospheric. If you're going to do data collection like I did on the tobacco at 0.5%, you should tell the instrument that before you collect the data. If this little introduction to how infrared gas analyzers work and dilution corrections is something that you find exciting. Amongst our vast collection of application notes at the Li-Cor website is an application note that talks in great detail about how ergas work and how we get dilution corrections. It even walks you through deriving volumetric dilution from the ideal gas law. It's very exciting stuff if you're into that kind of thing. Okay. So I want to talk now about how the instrument is architected, sort of how the flow path in the instrument works. And I want to start that by introducing this concept of a time constant, tau. I work with many engineers at Li-Cor, and I know over the years that my thinking has changed, and you know, I think in terms of tau, and I reference tau a lot because it tells us information about fundamentally how fast we can achieve things, how fast we can get our instrument to steady state, right? And tau is a function of volume and volumetric flow rate. So if we envision a perfectly mixed volume, a chamber has a lot of turbulence in it so that everything inside that chamber is at the same concentration. And we're feeding air into that chamber and we have air coming out the backside. And we change the concentration going in. Tau tells us how long the concentration coming out will be the same, how long until the concentration coming out is the same as what comes in, right? It tells us 1 times tau is 63% of the step change. We typically think in terms of 3 times tau, that's 99.5-ish% of the step change. We call that the full step change. So if you take volume of a chamber and we divide it by the volumetric flow rate, and then we multiply that times 3, that's sort of our fundamental limitation on how quickly we can do things. If we're considering a tube, tau changes meaning, but we compute it the same way. So for a tube, gas line, the time it takes a parcel of air to get from one end of that tube to the other end of that tube is a function of the volume of that tube and the volumetric flow rate through it. Okay? So understanding this tells us why we do things the way that we do at Li-Cor. And so I have 2 flow paths on the screen. One that I call a traditional open system. And this is every other instrument except for the ones that Li-Cor makes. It uses what's called a serial flow path, meaning things are in basically a straight line. Right? Air is brought into the instrument console where they measure the reference or the incoming. It goes through some flow meter so they know how much flow goes to the chamber. It goes through a long tube up to the chamber. It spends some time in the chamber based on tau. It goes through a long tube back to the console where it gets measured by the sample analyzer, right? So in a system like this, the total response time of the system is plug flow through this tube, 3 times the time constant for the chamber, plug flow through this tube. So we can't see anything that the leaf does faster than whatever that time is, right? And that time is generally tens of seconds. In a 6800, we bring air into the console and we just pass it through to the other side. We do some fancy environmental control in here. We'll talk about in detail tomorrow. And it goes up to the sensor head. In the sensor head, we split the total flow into 2. One side goes to the reference, one side goes to the sample, and then tightly coupled to the sample, or to the chamber, is the sample erga. And these flow paths are arranged such that our limitation in time response is really just this little chunk of the system right here, right? So when we open and close the chamber, we wait 3 times the time constant to stabilize in the chamber, and once we're stable, we can see anything the leaf does based on the time here. And at our maximum flow rate, that time is about 0.03 seconds for air to get from here to here. This gives us the opportunity to do fancy things with a 6800, like watch stomatal oscillations in real time. Okay. So let's talk a little bit about one of the most sort of critical things that we do for data quality with an open system. And if you think back to that mass balance equation that I showed, we're computing the carbon assimilation rate and the transpiration rate just from the difference in these two air streams, right? And we're doing that with two different gas analyzers. So we have one gas analyzer that measures sample, we have one gas analyzer that measures reference. When we take these gas analyzers and we think about them on paper, they seem like pretty perfect devices. When we take them to the real world, we find out that there's no such thing as a perfect device. They drift. They don't drift much, but if you're trying to measure deltas on the order of a couple of ppm, and they drift by a few tenths of a ppm, well, that might be a 10% error in what we're trying to measure, and so we need to account for it. And the way that we account for it in a 6800, or in a 600, as we'll talk about tomorrow, is by matching the ergos. And essentially what we do is we take, in the 6800, the sample airstream, and we vent it to the atmosphere, and we take the reference airstream and we split it between the two analyzers. And now the two analyzers seem the same air at the same point in time, and they should read the same thing. And if they don't, the instrument makes them read the same thing, right? We use this method of matching on the 6800 because it is operator proof. It doesn't matter the state of the chamber when we go into match mode. All that matters is that the airstream that would normally go to the reference analyzer is stable. And it's stable for 99% of most measurement conditions, right? There's a few cases where it's not. We'll talk about one of those in detail tomorrow, right? So it doesn't matter if I have a leaf in here, if my leaf is stable, if the chamber is open. This is one of the major technological advancements that came with the development of the 6800, was this form of reference-based matching. We have to think about when we match the ergos, right? Our general rules are every day before we start measurements, at least every 30 minutes, because we might have significant temperature drift over 30 minutes. Because we're in the field, temperature changes a lot in the field. When we're dealing with really low photosynthetic rates, we like to match carefully because small offsets, small errors could be big match offsets. And you can see match offsets aren't necessarily very big. In this case, when I matched this instrument, it corrected 0.03 ppm, sorry, 0.3 ppm. So anytime we change the concentration that we're operating at, and anytime we make a large change in flow. And we have two options for matching in the 6800. We have what we call point matches. That's the little red crosses that you see here. These are done at a specific concentration. And then we have something called range matching in the 6800 now. And that's where the instrument characterizes the match space. So it matches as a function of concentration. And it applies that match function instead of a fixed offset as it goes to make measurements. And range matching can be very handy for lab-based measurements because you can match once at the beginning of the day, and then you might not have to match ever again that day. So we can save time. For field-based measurements, where we're going to be in a place where temperature is changing a lot, we'd still probably want to do point matches. To give some sense of how much the match changes, I put my instruments, in this case it's a 6400 and a 6800, but what we're concerned about here is the 6800. I put these in a greenhouse in Li-Cor on a nice August day, and I just let them go and match themselves every couple of minutes, and I looked at the match offsets as temperature changes, right? And the blue line is the 6800. Over a 24-hour period, it drifts maybe half a ppm. It's not very much. You can see when it makes sort of sudden shifts, it's related to sudden changes in atmospheric temperature. And that if we're careful about these match offsets, right, We're just looking at delta CO2 down here, our proxy for assimilation. We can measure 0 pretty reliably. So I show this for a couple of reasons. The primary one is that as a scientist at Li-Cor, people often send me a data set and they say, Jason, would you look at my data and tell me if I'm using the instrument appropriately? Does this look reasonable to you? Generally new users. And myself and any other scientist at Li-Cor that gets this request, the first thing that we do is we go in the data file to this column called CO2 match. And we look to make sure that there's reasonable values there, right? We're interested to see that those values are not 0, meaning that the instrument was matched, that they're not some huge number. You know, they should be a few ppm, not tens of ppm or hundreds of ppm. And we're making sure that they're changing at reasonable intervals. Is the instrument being matched as appropriate in the field? I would encourage you, if you're evaluating somebody else's data, to do the same thing. Look at the match offsets and make sure they did the most important thing that you'll ever do with the instrument. So that gets us to 2 measurements from the 6800, ignoring the fluorescent stuff, right? We've discussed carbon assimilation and we've discussed transpiration, right? What I'd like to do now is talk about how we take those first 2 measurements and we make the next round of derivations to get the next set of physiologically relevant parameters from gas exchange. When we talk tomorrow, we'll go from this next round of physiologically meaningful parameters to a 3rd level, right? Where we're deriving those parameters in specific contexts that changes their meaning. But here, let's get through assimilation, transpiration, and then the next 2 interesting parameters. So when we measure assimilation and transpiration, this flux of carbon dioxide into or out of the leaf, or the flux of water vapor out of the leaf, it's important to note that that flux is fundamentally diffusive in nature, right? We're measuring CO2 as it diffuses from the atmosphere through the stomatal pore, through the intercellular airspace, where it moves across the mesophyll and onto the site of carbon fixation in the chloroplast. Along that same path, we have water vapor evaporating from the surface of the mesophyll cells, saturating this intercellular airspace, diffusing from this high water environment through the stomatal aperture out into the drier bulk atmosphere, right? That movement is transpiration, this movement is carbon assimilation. And these fluxes are diffusive in nature, and they share a common diffusion pathway. That means that we can redefine them following Fick's first law of diffusion. And Fick's first law just says that the diffusive flux of some gas, J, is equal to the diffusivity of that gas multiplied by its change in concentration over distance. This is the typical engineering formulation. To put it in physiological terms, things that might be more familiar to us, it says that the diffusive flux is equal to the diffusive conductance times the difference in concentration between 2 points in space. In the case of our leaf, we define these as the 2 endpoints of the diffusion pathway. So if we were to write these equations for transpiration and assimilation, following Fick's first law, we would say that transpiration is equal to the total conductance of water vapor along the diffusive pathway times the difference in water vapor inside the leaf minus that outside the leaf. And assimilation is equal to the total conductance of CO2 along the diffusive pathway times the difference in concentration outside the leaf minus that inside the leaf. And look at that, we've added one physiologically relevant parameter, this thing that we call CI, right? We'll talk about what this gets used for tomorrow when we give some specific measurement examples and how we do them. But for the moment, we can think about this as sort of a pool of carbon. It is the gaseous pool of carbon most readily available for the leaf to do photochemistry with, right? This is the carbon dioxide concentration in the intercellular airspace. And if we can start figuring out what some of these parameters are in this equation, that's how we get to solving for this thing. And the way we do that in these instruments, right? We've talked about how we get these measured fluxes, right? So we measure E, we start with water. We start by constraining water. We measure WA, right? That's the atmospheric concentration of water vapor. Well, that's really just WS, the water vapor concentration inside the sample. So we had to measure that to get E in the first place. Then, to get WI, we measure the temperature of the leaf, and we make the assumption that the intercellular airspace is at saturation at whatever that temperature is, such that if we know the temperature of the leaf, we know the water vapor concentration in the leaf, right? So now we know WI and we know WA, and we can solve for the total conductance of water vapor along the diffusive pathway. Well, this total conductance of water vapor is related to the total conductance of CO2 by their diffusivities in air. It's a ratio of 1.6. So now we've measured A. To get A, we had to measure CA, because that's really just CS, and we can solve for CI. So these equations are a simplified form of how it's actually implemented in the instrument. There's partitioning of these conductances based on stomodal distributions, and it gets a bit more complicated. But I like to present it from sort of this fundamental fixed law perspective, because I think these are handy troubleshooting and data quality tools, right? The first thing they tell me is that if I'm measuring a transpiration rate that's essentially 0, or an assimilation rate that's essentially 0, I can't really solve for CI with much confidence. So the smaller these numbers get, the greater the uncertainty in this value becomes. They tell me that, well, if I think about the mass balance, as long as my ergos are well matched, they don't have to read absolutely the right concentration, so long as when they see the same air, they read the same concentration. So it's easy to get A and E with good confidence. But now we're using absolute concentrations in this formulation to get CI. So it tells me that if I want to measure CI really well, then I care about the water vapor calibration on the instrument. I care about the carbon dioxide calibration on the instrument. It tells me that if I want to get CI, then I need to make sure I'm doing a good job of measuring leaf temperature. We give you a few different options for this on the instrument. One of them is to measure it directly with the thermocouple. The other one is to use a technique called energy balance. And we typically use energy balance for leaves that don't fill the whole chamber, leaves that are difficult to make the thermocouple contact. So, handy troubleshooting data quality tool. It's also useful in thinking about water vapor fluxes in the chamber. And I'm going to come back to this point in great detail. Before we get there, I want to talk about how we go from this parameter, total conductance to water vapor, to something more physiologically relevant, right? We don't care about the whole diffusive pathway when we talk about conductance. We really only care about the physiological part of that, the stomatal aperture. So how do we get from there, from here to there? So we apply what's called an Ohm's law analogy, and we borrow Ohm's law from electrical circuits, right? And Ohm's law says that in a resistive circuit, we can define resistances. This is the symbol for a resistor, the squiggly line, as existing in series with each other, right? These two are in series, or resistances that exist in parallel with each other. And Ohm's law gives us summing rules for how those resistances add to define the total resistance. And Ohm's law says that resistances in parallel, these guys, add as inverses, and resistances in series are additive. Now, I haven't talked about resistances yet. So, why do we use resistance? Well, resistance is the inverse of conductance. So 1 over resistance is conductance. 1 over conductance is resistance. So I said that resistances in parallel add as inverses. So conductances in parallel are additive. Resistances in series are additive. So conductances in series add as inverses. So we work in terms of resistance because it's an easy way to start. Okay? And why is that? Well, this is our full resistive diagram for the diffusive pathway between the chloroplast and the atmosphere, right? It says that there's a mesophyll resistance. There's a resistance contributed by the stomatal aperture. This resistance is variable. There's a resistance to diffusion across the cuticle, the cuticular resistance. There's a resistance to diffusion through the boundary layer. I'm going to come back to boundary layer in great detail in a few slides. And if I look at these, it's kind of complex. So we make some simplifying assumptions. Well, the first assumption that we make is that we're working with water vapor. So if we start our analysis of the total conductance from water vapor, mesophyll resistance just disappears, right? Because water doesn't evaporate as a gas across the mesophyll. It doesn't diffuse across the mesophyll as a gas, right? It evaporates from the surface of the mesophyll. So there's no mesophyll resistance. It goes away. And then we make the assumption that cuticular resistance is near infinite. Well, what's 1 over infinity? It's 0. It goes away. And we're left with a simple model that says we can take the total conductance that we measure and we can figure out something about the stomata if we know something about the boundary layer outside the leaf, right? It just says that 1 over our total minus our boundary layer is stomatal conductance. Easy, sort of. So the boundary layer is a layer of still air that builds up at the leaf surface, right? And I have a simplified diagram of one here. And I present this because it comes from maybe one of the most useful texts if you want to really delve into the biophysics of what we do with these instruments and how we derive the parameters that we measure. And this is Park Nobel's Biophysical Plant Physiology and Ecology. There's a whole bunch of different versions of this thing that have come out over the years. But this model is for a planar surface with air approaching it from one direction. And it says that the thickness of the boundary layer, which is directly proportional to its resistance, so the thicker the boundary layer, the greater its resistance or the lower its conductance, is a function of how fast that air approaches the surface and how wide that surface is, right? And so the wider the surface, the slower the air, the thicker the boundary layer becomes. And this happens because that surface produces drag on the air. And so the air up here that might be able to move very fast because it's sliding past the relatively spaced out air molecules. And as we get closer to the surface, it's getting caught on the roughness of that surface. It's being dragged across it, and it's becoming progressively slower and slower and slower. Boundary layer is a really important concept in gas exchange measurements because we actively manipulate it in the chamber. If we think about a leaf out in the bulk atmosphere, that leaf has some boundary layer around it. If its stomata are open, they're connecting the intercellular airspace, which is completely saturated, to the air outside the leaf in that boundary layer. This layer of still air means there's very little turbulent exchange between the bulk atmosphere and the leaf surface, so that a gradient in humidity gets established in the boundary layer. Our leaf out here in the bulk atmosphere has a humidity near its surface that's pretty close to saturation, and a humidity out at the edge of the boundary layer that's near whatever the ambient is. You probably experienced this the consequences of the boundary layer. You probably have experienced the consequences of the boundary layer. At some point, you know, you've gone out in the evening and there's a grassy lawn, and you're out by the grassy lawn, and as the sun goes down, there's still some light in the sky. You notice that the grass is nice and dry, it's nice and dry, and then as the sun dips below the horizon, all of a sudden, there's dew all over the grass, right? And that dew formed because when the sunlight stopped shining on the grass, it cut the radiative load. There's still some transpiration going on, so it's dropping energy out. That drops the temperature of the grass blades. They were setting in an environment that was already very close to saturation at whatever temperature they were being driven to by the sun. And now that that radiative load is gone, they get below it and water condenses out, right? That's because of the boundary layer and this buildup of water at the leaf surface. When we put a leaf in the chamber, we have this thing called the mixing fan or the leaf fan, and we use it to provide just a tremendous amount of turbulence around the leaf, right? And that tremendous amount of turbulence makes the boundary layer at the leaf surface very, very thin, right? Smaller the boundary layer, remember, the greater its conductance. And the inverse of the conductance is its resistance, right? So the lower the greater the conductance, the lower the resistance. Because the resistance is very small, we work in terms of resistance. Its contribution to the total becomes small. And so we don't have to know it particularly well to be able to partition it out and get at just the stomatal contribution. That's not to say we don't know it. We characterize it very carefully as a function of leaf geometry, chamber geometry, fan speed in every one of our chambers. And we model it on the fly to correct for it. But we're actively manipulating it. The consequence is that when we put a leaf in the chamber, that leaf is experiencing an environment in the chamber that's very close to whatever the chamber humidity is. So if we make our chamber very dry, then the leaf is seeing something pretty close to that dry humidity. If we make our chamber very wet, it's seeing something close to whatever that very wet humidity is. This is important in thinking about how we configure the environment in the chamber. It means that when we go to make measurements, we want to target boundary layer conditions. I think everybody has their own metric of humidity that they feel comfortable thinking in. For me, I like to think in the context of a VPD, so a vapor pressure deficit between the leaf and the atmosphere in the chamber. Lots of people think in terms of relative humidity. Whatever you do, you want to choose the humidity such that the chamber is pretty close to what the boundary layer was. For most measurements, we would say that you want to use a VPD somewhere between 1 and 1.5 kPa. That's a rule of thumb. It's not an absolute. It's not an absolute, right? But generally, for most plants, if we increase VPD above 1.5 kPa, we've made the chamber dry enough that will induce a stomatal response. And so by putting the chamber on the leaf at that sort of vapor pressure deficit, we'll make the stomata start to close. If we go well below 1, eventually we approach the dew point and we reach condensing conditions. And our stomata might stay open, but we'll make it rain in the ergas, and that's not very good. Now in Nebraska, this is easy to think in terms of because, well, it's not very wet here. In Indonesia, it's pretty humid, right? And so you think, well, what's 1.5 kPa? Well, that's like 55% relative humidity. Well, what's 1 kPa? It's somewhere around 60, 70% relative humidity. which is probably a little bit below whatever your atmospheric is. So where you are, you might have to work at slightly higher humidities. The way that I would evaluate this is I would start somewhere around 1 kPa. I'd put a leaf in a chamber at a light intensity that it's already used to and see what happens with the stomata. If they start to close, I'd make it a little bit wetter. And I would keep making it wetter until the instrument tells me that I shouldn't do that. It'll give you an error warning that you're approaching condensation. Interestingly, for tropical plants, there is a case where you can make it as wet as you want in the chamber, and the stomata will always close the moment you put the instrument on them. And that's for broad-leafed tropical understory plants and broad-leaf tropical trees. There is a known stomatal response to turbulence, right? So the fact that we have a fan in there stirring the air up, dropping that boundary layer around the leaf, there's some plants that'll respond to that. I've measured a lot of different things in my life, but I've only ever seen this twice. I saw it once in measuring Theobroma cacao. We were working with greenhouse-grown plants at the Beltsville, Maryland USDA facility. And no matter how wet we made it, those stomata closed. And then we started turning down the fan speed, which you should only do if you know you need to do it. And this was one of those cases. And we saw that the stomata started to open back up at the same humidity. And that's a good indicator that it was a turbulence response. The other place I've seen this is in Amorphophallus. It's a giant aeroid that grows in the tropical understory in Sumatra, I think. And it has a huge leaf with a massive leaf area, and it grows in an environment at fairly low light and very high humidities. And it was very sensitive to turbulence. So I want to circle back to this fixed law thing. And I want to discuss sort of the context of, or put the measurement of water vapor from the instrument in sort of two contexts, right? Or the two measurements. So we've measured this thing that we call transpiration. And I would argue that the right term for it is called apparent transpiration, or E-apparent. And then we've talked about this thing that we call stomatal conductance to water vapor. And maybe the first thing to note here, and you can see it from the approximation from fixed law on the screen, is that stomatal conductance and transpiration have the same units. They're both in molar flux, right? Moles per meter squared per second, right? Or millimoles per meter squared per second, depending on how you want to scale it. Fundamentally the same thing. So we put a leaf in a chamber, and that leaf starts to release water vapor, and that's the apparent transpiration rate. And when we measure that in the instrument, we're absolutely counting the number of molecules that come out of the leaf. That is the real transpiration rate in the chamber. But if we think about transpiration from fixed law, well, what is transpiration? Well, it has a physiological component to it. That's how open the stomata are. And it has a physical component to it. That physical component we sometimes call evaporative demand, right? It's that diffusion gradient in water between inside the leaf and outside the leaf, right? That difference between Wi and Wa, right? So physiological stomatal conductance, physical Wi minus Wa, evaporative demand. And I talk about this because this E value, I call it E apparent, is subjective to measurement conditions. So I think an example helps a lot here. So I have two plots on the screen. This top plot shows transpiration as a function of leaf temperature in blue, and as a function of chamber humidity in green, right? And this is data that I just modeled. So I took that approximation on the screen and I put it in Excel and I held the stomatal conductance constant, right? I didn't change it. I just changed leaf temperature So changing WI, or I changed humidity, WA, right? And we can see that as we change one of those parameters or the other, it causes a change in the apparent transpiration rate in the chamber. And if you were to back calculate my VPD standards to relative humidity, you'd see that sort of nominally we're talking about 50 to 70% relative humidity. And if we set the chamber up to be in that 50 to 70% relative humidity range, without changing the stomatal aperture, we can drive transpiration over a twofold range, right? We can double it between one end of that humidity and the other end. And I presented this to a friend at Brookhaven National Lab, and he said, Jason, you can make Excel do anything. Prove to me that a real leaf actually behaves that way. So I went back and I took a capsicum, a bell pepper again, and I got it nice and stable in the chamber. And I just changed the humidity up and down. This is randomized. And you can see over my range of operating humidities where I would normally make measurements, right? There's no change in stomatal conductance. It's constant. That's the blue. But that transpiration rate that I measure changes dramatically. Twofold. All from changing the humidity in the chamber. So I present this because, you know, we have these two metrics of water vapor leaving the leaf. We have the transpiration rate and we have the stomatal conductance. My preference when I report a water vapor flux from an instrument, from a chamber-based measurement, is to always report stomatal conductance. Because fundamentally, it is the transpiration rate standardized by the diffusion gradient, right? It makes it really easy to make apples to apples comparisons between two measurements, right? It doesn't matter if the leaf temperature was a little different in the first measurement from the second measurement, or I was at 50% humidity for the first one and 70% humidity for the next one. When I compare stomatal conductance, I've leveled that playing field. They're directly comparable. When I compare transpiration rates, I really have to think about the physical environment they were measured under. And it's difficult to make direct comparisons of transpiration unless I controlled that physical environment really specifically. This is important in the context of things like water use efficiency, right? So it's very tempting to take gas exchange data from a chamber and compute instantaneous water use efficiency. Lots of people do it. If I were going to do it, I would suggest the alternative metric of using intrinsic water use efficiency, right? And that's carbon assimilation divided by stomatal conductance. I like intrinsic water use efficiency because it accounts for evaporative demand, dA is WI minus WA. And because in C3 plants, it's the same physical process that allows us to derive carbon isotope or derive water use efficiency from carbon isotope data. So if I take a C3 plant and I grind it up and I look at its isotopic ratio, I can get water use efficiency from that. That water use efficiency is because there's a fractionation event that happens across the stomatal aperture. And so when we look at it from stable carbon isotope data, we get water use efficiency integrated over the developmental history of the tissue. When we look at intrinsic water use efficiency, the same physical process, we can look at it at a moment in time by taking A and dividing it by GS. So I think I'm running ahead on time, so we'll have room for lots of questions at the end. I think I'm used to more interruptions when we do this. So hopefully we'll have lots of interruptions to go through. Okay, so let's talk a little bit about hardware. In a couple of slides, I'll show you some of the different accessory tools we make to the 6800. But fundamentally, these things are all chambers, right? There are parts that are constant to the instrument. That's the console. This is what houses the batteries that power everything, the computer that controls everything, the air conditioning supply, what allows us to control CO2 and water vapor in the system. It gives us the user interface and the data storage. There's the sensor head, right? This part with the handle on it. That's where the ergas are housed. That's where the temperature control happens. And then there's the chamber. This part right here. And in this case, this is the MPF fluorometer. And the chamber is where we would clamp on a leaf. Right? You can see the leaf aperture here. And in the case of the fluorometer, it contains both the PAM fluorometer and a light source. So we also make chambers that are just light sources and an aperture. They don't have a fluorometer in them. And there's a difference in size between these various apertures, right? So in the fluorometer, the smallest one is 2 square centimeters. The one shown here is 6 square centimeters. And then when we go to chambers that are just light sources, those become up to 9 square centimeters, up to 36 square centimeters, right? So the aperture is becoming bigger and bigger and bigger and bigger with different chambers. And if you think back to the mass balance that I presented, right, what did I say? I said that for any given assimilation rate, if we change the amount of leaf area that's in the chamber, so we put more leaf area in the chamber, or we take leaf area out of the chamber, right, we change how big the delta CO2 is, the signal that we're measuring. So if I have a 2 square centimeter chamber and 20 square centimeter chamber, pick that because the math is easy, and I put the same leaf in those 2 chambers, and I measure the same assimilation rate, in that 2 square centimeter chamber, the signal that I'm measuring will be 10 times smaller than it is in the 20 square centimeter chamber. It's a lot easier to measure something that's 20 times bigger than it is to measure something that's 20 times smaller. So we generally say that when we're going to go make measurements, we'd like to use the biggest chamber we possibly can. Right? The bigger the chamber, the more leaf area, the more signal we have to work with. And this inevitably raises the question of, well, what if my leaf doesn't fill the bigger chamber? Right? Lots of leaves are smaller than even a 2 square centimeter chamber. And here I would say that you still want to use the biggest chamber possible. It doesn't matter if the leaf entirely fills it. What matters is that if you don't entirely fill the chamber, you tell the instrument how much leaf area was in there. You can do that through the user interface as you're collecting data, or you can do it in the data files after the fact. The data that comes out of the instrument is an Excel file. It has all the equations built into it. There's a column for leaf area. And all you need to do is type the actual leaf area in there, and everything that's dependent on it will recompute. The only time a smaller chamber helps you is when your leaf is going to fill it completely, because then you know exactly what the leaf area is, and you can get through measurements quicker, right? Because you don't have to go back and measure it. The next consideration that I make when I'm picking a chamber is, does it have a light source? My preference is always to use a light source, right? The sky is tricky. I'm going to show some data tomorrow measured under different sky conditions. And we'll talk about how small changes in leaf angle can have dramatic effects in the amount of light that a leaf is seeing. And for me, if I use a light source, something where I can type in the light intensity that I want, I know really well what the light intensity at my leaf is. It's not subject to how I hold the instrument. It's not subject to little clouds passing over that might change things, right? It's under my control. And so my data is inherently more constrained. And then the next thought is, well, what data do I actually need? Do I want just gas exchange data? Sometimes that's okay. Lots of times I want gas exchange and fluorescence data. But if I want fluorescence data, then my only option is to use a light source that includes a fluorometer. And that would be this one here. So this is kind of how I think about it for a leaf level measurement, where I'm going to go clamp on a leaf. But there are other kind of measurements that we make with these instruments. And Li-Cor makes a whole bunch of different hardware that you can use to measure different types of materials. And so these are examples of sort of typical clamp style chambers. As I was talking about, like with the fluorometer, we're going to go clamp on a leaf and make a measurement. So this is the 3x3 chamber. It's 9 square centimeters. This is the 6x6 chamber. It's 36 square centimeters leaf area. So both of these chambers are either clear topped, or you can use them with a light source. There's a red-blue light source for this one. This chamber uses the light source shown here, which is an RGBW component light source. So it has red LEDs, blue LEDs, white LEDs, and green LEDs. And they're all independently controllable. So we use this light source when we want to explore spectral response of photochemistry, right? Because we can make measurements of photochemistry under monochromatic light or various mixes of light. And that goes on this chamber. The other thing that we use this chamber for is if we want to make measurements using some third-party analyzer. So on the 6800 sensor head, there's options to connect it to analyzers that measure gases that aren't CO2 and water vapor. So we have a nice application note that documents how to take this chamber and a laser-based instrument to measure oxygen flux from the leaf, right? You can take that same approach, and there's a number of labs that do this, and they couple it to laser-based instruments to measure carbon isotopes. And so you can take this 6x6 chamber and measure isotopic fractionation on the fly. Okay? Or we have an example in Works where we take this chamber and we couple it to an analyzer that we make that measures methane, and we use it to make direct leaf level measurements of methane. And one of the neat things about the 6800 is because we can control the pressure in the chamber and the humidity in the chamber, that potentially can be used to tease apart the mechanisms of methane transport in plants, right? Whether it's coming through some air-filled passage or it's being transported by gas dissolved in the transpiration stream. We make chambers that are used for discrete samples. This is the bryophyte chamber. This is the Arabidopsis chamber. We use it to measure whole canopies of small rosetted plants like Arabidopsis grown in containers. The bryophyte chamber we use to measure discrete samples of things that could be like a fruit. It could be something like a clump of moss. We use these to get carbon fluxes. We make chambers for the 6800 that you can take to the field and you can measure carbon dioxide evolution out of soil. So this is the soil chamber for the 6800. We make chambers that people use to measure small discrete samples that don't require light. So we call this little plastic tube arrangement here the insect respiration chamber. And it's used to measure insect respiration or respiration from small fruits or seeds. And then people build their own chambers that are interfaced to the 6800. We would call this a custom chamber application, and we provide the manifold back here for the interface. And then people build a chamber like this clear one shown here. And it's nicely photographed with an apple in it. But when we validated this, we took it to the field for the application note to do canopy level light response on turf. Before I came to Li-Cor, I spent a lot of time building custom chambers to measure photosynthetic carbon assimilation and transpiration from orchid roots. Orchids have photosynthetic roots. They don't really fit in any sort of chamber that Li-Cor makes. And so I used to build my own chambers. The one thing to note about these is my whole discussion of stomatal conductance and CI is predicated on diffusion across a plane. And so when we go to measure something that's a complex canopy, like an Arabidopsis plant or a clump of moss, right? And we think about how we derive stomatal conductance and CI. If you think about that leaf temperature measurement, well, what's the leaf temperature in a canopy, right? There's certainly variability in it because it's not all at one uniform light intensity. It's not all of uniform turbulence. Well, if it's not at uniform turbulence, then what's the boundary layer really in here? And so generally when we make these measurements in these kind of chambers, we're just reporting a carbon assimilation rate. So I think now that we should take a couple-minute break, and then we can come back and I will spend the rest of the time answering questions that anybody may have. When we reconvene tomorrow, I will set an instrument up to make a measurement, and I will let it make a response measurement as we talk about sort of the different kind of ways that we apply the data and how that changes the meaning of the values and the different things that we can derive from the instrument. And then hopefully that goes well. We'll learn about diurnal rhythms if we don't already know about those. And that might mess us up. And then after that, we'll talk a bit about the new porometer fluorometer and how it works. And I'll give some example data sets and some example data sets that show how it pairs with the 6800. Okay?
Okay, Jason. Thank you for your presentation. Now the session of question and answer. But for you, maybe take a rest for a while, and we will answer the question for Professor Kukuh. Okay?
Okay.
Okay, ya. Profesor Kukuh. Halo, Pak. Pak Prof. Halo.
Ya, jadi kalau saya jadi sebentar pergi kan, nanti jam.
Ya. Sorry, dari toilet. Gimana?
Jadi kita masuki sesi tanya jawab, Pak.
Ya.
Untuk yang sesi awal ini mungkin pertanyaan untuk Bapak.
Yuk.
Untuk khusus untuk Jason, Pak. Prof, ini ada beberapa 4 pertanyaan. Yang pertama, bila mungkin terlewat, bagaimana cara mengamati respirasi akar?
Ya. Terus?
Itu aja dulu, Pak. Nanti yang.
Oke. Satu dulu ya.
Ya, Pak. Ada 4.
Oke. Terima kasih. Terima kasih, Pak Mamen.
Ya.
Ini dari pertanyaan bagaimana cara mengukur respirasi akar.
Ya.
Jadi
apa nih?
Kalau mau diekspor, Pak Kukuh itu. Ya. Halo, Pak.
Ya, Pak.
Silakan.
Yo.
Silakan,
Saya
Pak.
jelaskan ya.
Ya, silakan, Pak.
Ya. Jadi cara mengukur respirasi akar yaitu tanaman dicabut dengan hati-hati dengan air yang mengalir pelan-pelan ya. Jadi air tidak keras, air gently ya, Pak ya. Hati-hati sekali disiram. Dan tanaman ditanam di dalam pot atau di lapangan. Jadi setelah digali itu pelan-pelan disiram, dihilangkan tanahnya. Setelah hilang tanahnya semua, dipotong, langsung dimasukkan ke tabung. Nah, ada tabung ukuran besar, kecil ya. Dan itu ditutup rapat secara langsung, jadi tidak usah menunggu dengan seal. Nah, setelah itu diinkubasikan. Ya, ada yang 2 jam, ada yang 3 jam, ada yang 4 jam. Ya, oke 3-4 jam. Kalau setelah itu nanti akan jadi metan. Yang kita ukur adalah CO2-nya. Nah, itu nanti setelah itu suntikan itu ditasukkan terus dipompa supaya sirkulasi di dalam tabung itu CO2-nya rata. Pompa terus ditarik ke siring, diambil terus dibawa disuntikan ke alat infrared analyzer. Nah, jadi dia akan disuntikan situ, dia akan terbaca kandungan CO2-nya. Itu dia respirasi akar. Jadi berapa CO2 yang dikandung situ nanti berdasarkan bobot akar. Setelah dianalisis, bobot akar dikeluarkan terus ditimbang. Itulah dia. Nah, jadi CO2 per gram atau per miligram tentang bobot akar. Itu Pak Memen. Jadi jangan sampai nanti membersihkan akar dari tanah itu dengan air yang deras, jangan air yang gentle ya. Biasanya saya pakai streaming saya tutupi akar itu streaming terus saya siram pelan-pelan supaya dia menekan pelan-pelan dan tidak langsung air itu tidak langsung tumpah kepada pembersihan tanah, tapi dia langsung ke streaming dulu terus baru pembersihan akar. Itu Pak Memen.
Baik Pak Prof.
Ya.
Berikutnya yang kedua pertanyaan. Tadi dikatakan mengukur fotosintesis secara tidak langsung dapat menggunakan berat segar dan berat kering tanaman.
Ya.
Secara kuantitatifnya, bagaimana laju fotosintesis didapat dari berat tanaman? Karena parameter ini sering ada dalam penelitian-penelitian, tapi jarang dikaitkan dengan laju fotosintesis. Nah itu Prof, pertanyaannya.
Yuk, terima kasih. Memang sekarang kan memang ketersediaan alat kan nggak ada. Kecuali kalau di Universitas Lampung ya mungkin salah satunya ada, itu bisa langsung fotosintesis. Yang belum ada itu biasanya ada 2 pengukuran fotosintesis, yaitu secara langsung dan tidak langsung. Kalau langsung ya dengan alat Li-Cor, dia menggunakan sirkulasi udara di situ ya. Kalau yang tidak langsung bagaimana? Apakah bisa? Tetap bisa dengan biomas, bobot biomas. Gitu. Jadi sudah terukur sini bahwa di mana-mana penelitian saya juga menunjukkan bahwa laju fotosintesis berkorelasi positif terhadap bobot kering atau bobot basah tanaman. Jadi semakin bobot biomasa tinggi maka laju fotosintesis tinggi. Itu harus mengarah ke situ dan kalau mahasiswa nulis harus ada reference dari mana dia mengatakan bahwa fotosintesis tinggi, biomasa tinggi. Itu harus ada referensinya ada. Itu juga sudah banyak referen di mana-mana, tinggal masih itu. Jadi memang bagi yang belum ada alat fotosintesis, ya bisa menggunakan biomasa, Pak Memen, gitu Pak Memen.
Baik. Untuk pertanyaan ketiga, Prof.
Yuk.
Indikator biokimia yang digunakan untuk mendeteksi toleransi tanaman kacang tanah terhadap cekaman kekeringan adalah ABA, EBA, dan sitokinin. Mengapa Prof Kukuh tidak menggunakan prolin? Mohon informasinya. Mohon juga informasinya kaitannya dengan metode pengukuran kandungan EBA dan sitokininnya. Itu Prof.
Yo, terima kasih. Wah ini agak panjang ini ya.
Atau gimana?
Jadi ya, nggak apa-apa, oke ya. Ya pelan-pelan ya. Jadi gini, jadi memang prolin selama ini ada orang menggunakan sebagai indikator cekaman. Entah itu panas, entah itu suhu tinggi, atau entah itu drug, entah itu penyakit, itu cekaman. Begitu cekaman, dia langsung prolinnya tinggi. Nah, saya menggunakan juga sebelum ke ABA sama sitokin, saya menggunakan juga prolin. Saya sudah berusaha untuk men-screening varietas bukan sebanyak itu, tapi beberapa itu yang terpapar kekeringan. Maka dia itu ada yang tahan kering, prolinnya tinggi. Ada yang tahan kering, prolinnya rendah. Nah, begini. Begitu di lapangan dia itu tahan kering, karena berdasarkan prolin, saya tanam di pot. Nah, rupanya berubah dia. Dia tidak tahan drug pada saat di pot, yaitu 3,4. Jadi, waduh, inilah yang membingungkan saya bahwa prolin bisa digunakan secara cepat, ya, bahwa dia itu kekeringan. Tapi yang jelas EBA dan sitokinin, rupanya dia fungsinya ada. Kalau EBA itu adalah dekat dengan pengaturan stomata, buka tutup stomata. Sehingga begitu akar terpapar kekeringan, stres, maka akar kan berkomunikasi ke daun untuk menutup atau membuka sedikit stomata. Sehingga tadi yang tahan kering, EB-nya tinggi di daun. Karena dia itu. Kalau yang varietas yang tidak tahan, dia tidak bisa berkomunikasikan antara akar dan daun. Sehingga kandungan EBA atau apa di daun rendah. Akibatnya ya tidak bisa tidak bisa mengontrol stomata buka, transpirasinya naik. Lah di sini mengalami kekeringan dieback of leaf terjadi itu. Oleh karena itu maka indikator kekeringan tadi itulah yang saya gunakan apa. Jadi ada konektivitasnya ya di situ. Dan sitokinin juga gitu. Begitu sitokinin itu mampu di daun, maka dia retensi klorofilnya terjamin. Apa yang dibutuhkan fotosintesis, maka disitulah disediakan sitokininnya. Dan dia itu retensi klorofil terjamin tidak menurut drastis seperti yang tidak tahan kering. Nah, ini dua itu yang mendukung penuh. Yang EBA pengaturan stomata dia itu, dan yang sitokinin dia itu pengaturan klorofil retention. Jadi itu yang Pak Memen. Nah, yang analisisnya itu, analisis untuk EBA adalah pengambilan sampel daun, ya, daun. Ya. Silakan daun muda bisa, daun tua, tapi jangan kering lah, daun segar. Ya. Jadi itu diambil 5 gram. Sekarang diambil 1 gram cukup. Nah, itu dulu saya 5 gram. Karena memang takut kekurangan itu ya. Sekarang 1 gram cukup. Nah, itu nanti di homogenizer dia dengan etanol 70%, terus disaring dengan microfilter, terus nanti diuapkan homogenizer etanolnya diuapkan maksudnya, itu aqueous itu dimasukkan ke dalam tabung deteksi saring. Ya, di situ. Jadi itu menyaring polar-polar yang tidak berfungsi untuk pengukuran EBA. Nah, setelah itu nanti diambil melalui siring atau disuntikan ke SPLC. Ya. Jadi SPLC suntik, terus kita pakai internal standar EBA asli. Nah, itu ada pada retention time antara 6 sampai 7. Ada yang 5, ada yang tergantung nanti internal standarnya. Di sini saya gunakan 6 sampai 7 retention time. Nah, setelah itu saya tampung di 4, 5, 6, 7, 8 tabungnya. Jadi ketahuan retention time 1 tabung 1, retention time 2 tabung 2. Itu ada putar di tampung airnya setelah mengalir di SPLC saya tampung. Nah, di tabung itu saya murnikan lagi. Nah, setelah saya murnikan lagi, maka saya langsung ke ELISA. Jadi situlah pakai antibodi segala bahwa itu benar-benar EBA yang saya ukur, bukan EBA kuantitas. Jadi EBA yang saya ukur betul, itu udah ELISA. Itu ya, jadi sangat mudah dan cepat sekali. Ya, cuma ada beberapa tahap. Jangan yang sitokinin itu juga saya pakai kertas kromatografi. Jadi dipisah gradien. Ya. Setelah homogenizer seperti biasa 70% etanol, terus diuapkan, disaring seperti tadi, terus tadi saya pakai pemisahannya dengan kertas kromatografi. Saya gunting 1 cm sampai ke atas. Terus dari saat itu saya larutkan, cairkan, terus saya seleksi nomor berapa kira-kira ada sitokininya, sama seperti tadi ya. Setelah itu setelah ada nomor sitokinin itu ada di daerah 13 ke atas ya. Jadi kertas kromatografinya panjang sampai ke atas. Itu nanti setelah saya ekstrak lagi dalam bentuk aqueous, terus saya semprotkan ke biji mentimun. Nah, saya masukkan ke dalam tempat gelap. Nah, biji mentium itu gelap, dia bercambah, jangan diekspos cahaya, berbahaya, karena itu nanti akan klorofil ada. Setelah diekspos gelap, kecambah dia, maka saya langsung ekstrak kecambah tadi, kecambah mentimun, terus saya ukur dengan spektrum fotometri dengan 665 nanometer. Nah, itulah dia sitokinin endogenus. Jadi mudah-mudahan nanti kita kalau mengenai itu bisa diskusi lagi itu. Terima kasih Pak Memen. Halo, Pak Memen. Halo. Halo, kok mati? Anunya mati mau ngomongnya. Suaranya nggak kedengaran.
Halo.
Nah,
ya Pak.
Yo, ya.
Oh, ya. Pak. Tadi itu pertanyaan dari Bapak Muhammad Suardi dari Universitas Hasanuddin, Pak. Pertanyaannya. Untuk yang pertanyaan terakhir, ini dari Pak Supartoto, Pak, Universitas Setiawan. Pertanyaannya adalah, hasil persilangan Bapak tadi belum menghasilkan F1 dengan jumlah klorofil lebih tinggi dari parensnya. Apakah mungkin keturunan dengan klorofil lebih sedikit berproduksi lebih tinggi? Itu Pak pertanyaannya. Terima kasih.
Oke, ini Pak Supartoto ini teman nih Pak.
Oh.
Teman UNS. Terima kasih Pak Supartoto. Jadi yang kami tadi dari sekian komponen fotosintesis dan fungsinya rupanya yang menonjol adalah stomata. Jadi saya harus putuskan bahwa stomata ini diwariskan enggak? Karena stomata itu adalah yang Jason jelaskan tadi adalah tempat pertukaran air dan gas di situ sangat penting di situ. Sehingga itulah salah satu komponen fotosintesis yang menonjol. Oleh karena itu saya berusaha untuk apakah diwariskan nggak ini. Nah, kalau sudah begitu maka memang betul bahwa laju fotosintesisnya tinggi dengan adanya stomata tadi, tinggal dia itu karena diwariskan tadi. Ya. Dan memang kalau klorofil memang belum dihitung di sini. Ya, kalau memang diteliti, ini salah satu peluang bahwa sudah ada stomata diwariskan. Memang klorofil diwariskan, tapi secara real dihitung masih belum. Jadi silakan nanti teman saya Pak Supartoto nanti kita bisa membuat proposal sama menghitung Li-Cor sekalian apakah memang klorofil rendah, klorofil tinggi, bisa nggak dia itu fotosintesisnya tinggi atau rendah. Dan jelas klorofil diwariskan, cuma yang ukur tinggi atau rendah memang belum. Walaupun secara teori kalau klorofil rendah pasti fotosintesis rendah, itu secara teori. Tadi saya nggak bilang itu menjamin bahwa itu. Barangkali ada yang lain lagi kita bisa salah nanti itu ngomongan itu. Jadi itu dia Pak Supartoto. Saya menunggu untuk sekalian penelitian bersama. Mumpung ada Li-Cor 6.800 ini dimanfaatkan sebaik-baiknya dan kita nanti hitung klorofil diwariskan nggak dengan turunannya betulnya. Itu Pak. Pak Memen. Terima kasih Pak Memen.
Baik Prof. Pertanyaan terakhir Pak.
Yuk.
Dari support service LI. Pertanyaannya, kapan sampel daun dan akar diambil untuk dapat dianalisa kandungan hormon EBA dan sitokinin supaya kita dapat membedakan respon tanaman terhadap stres? Itu Prof pertanyaannya terakhir.
Oke. Ya, terima kasih. Memang bagus pertanyaan ini, terima kasih. Jadi waktu awal itu memang sangat tricky sekali di sini ya. Jadi begitu stres langsung akan berkomunikasi. Tapi kita juga hati-hati sekali bahwa kapan kita harus menganalisis itu. Nah, di sini saya analisis yang paling sesuai itu adalah saat pengisian biji. Jadi di situ EBE akan bermanfaat di samping pengaturan stomata, dan dia akan bermanfaat juga selama laju pengisian biji. Sehingga semakin tinggi EBE, semakin tinggi pengisian biji. Itu hubungannya ke situ. Jadi kita tidak bisa awal bahwa mengukur EBE tinggi, ya nanti kalau hasilnya tinggi. Kalau enggak, nah itu langsung pengisian biji. Itulah dengan adanya EBE tinggi, maka dia hasil pengisian biji tinggi. Nah, itu dia. Jadi saat-saat pengisian biji lah itu yang tepat untuk pengukuran EBE dan sitokinin, Pak Mimin. Terima kasih. Ini bagus pertanyaannya.
Baik, Prof. Terima kasih. Semua dijawab pertanyaannya. Untuk berikutnya mungkin pertanyaan khusus buat Jason, Pak. Ya, terima kasih, Prof.
Ya, semoga balik. Terima kasih kembali.
Halo, Jason. Are you there?
I'm here.
Okay.
I'm ready.
Your turn now. We have 3 questions for you.
Okay.
Yes, okay. The first is biologically plant growth is determined by C gain or carbon gain from assimilation and C loss from respiration. How can Li-Cor translate this physiological process? This is the question for you.
Yeah, that's a good question. I mean, it's sort of fundamentally what we measure with the instrument. So when we do the gas exchange measurement for carbon dioxide to measure assimilation, we're measuring the movement of carbon into or out of the leaf, right? And we make that measurement in a few different ways. So one of them is that we measure in the dark. And if we measure carbon flux in the dark or carbon assimilation in the dark, what we'll see is carbon coming out of the leaf. And that rate of carbon coming out of the leaf is an estimate of the dark respiration rate. It's not necessarily the respiration rate in the light. There are techniques to use the Li-Cor to get at that, but it's not quite as direct. And then we make measurements of that carbon assimilation rate under instantaneous conditions, what we'd call a survey measurement or a point measurement of what the leaf is doing under its current environment at a point in time. We might make that measurement repeatedly over time to build a picture, or we make that measurement under different light intensities or different carbon dioxide concentrations, what we'd call a light response or a CO2 response curve. And when we want to understand the Li-Cor measurements in the context of total carbon gain, total carbon loss, generally we take those response measurements and we'd use a physiological model to relate what we get from the response measurement to how the leaf or the whole canopy might behave.
Okay. We move to second question. Sorry. One moment. Sorry. Waiting.
No problem.
Okay. Okay. Second question is, how can Li-Cor explain the relationship between photosynthesis and leaf area index?
Okay. So, well, firstly, when we measure photosynthesis, the carbon flux or parameters related to it like stomatal conductance, these things are expressed on an area basis, right? So there's explicitly a relationship between leaf area and the number that we report. If we're thinking about leaf area in the context of taking, say, a measurement on a point on an individual leaf and trying to scale it up to what a canopy might be doing, then that's another place where we would take response measurements. So measurements of either response to light or response to CO2, or the combination of those, and we would apply them in a model and try and scale up. So light changes through the canopy, CO2 concentration changes through the canopy. We have to account for how that works. It's not as simple as taking a point measurement on one spot and multiplying it by the total leaf area for a given area. That's what's called a bottom-down approach. We didn't talk about the instruments today, but Li-Cor also builds instruments that are used for top-down approaches to relating photosynthesis to sort of total leaf area. And these instruments are, they use a technique that we call eddy covariance. It's a micrometeorological technique where we measure carbon dioxide and water vapor at a point up in the sky above our canopy, and we measure the three-dimensional wind speed at the same time. And eddy covariance relies on this idea that as wind moves across the eddy, it's moving in sort of a circular motion that's bringing air from the bulk atmosphere down to the canopy where it interacts with the leaf, and then it's bringing that air back up. And so if we have an actively photosynthesizing canopy and we measure the air that's going down, it will be higher in concentration than the air we measure coming up. And by measuring the three-dimensional wind speed and the concentration in that air, we can characterize those eddies, and we can compute the carbon assimilation rate or the carbon loss rate for... So at that point, we're making measurements not over a little tiny leaf area, but we're integrating over, say, an entire field or an entire forest, and then relating that back to leaf area from the other way.
Okay, the next question. Since the measurement based much on gas exchange, will the microclimate around the plant, especially wind velocity and air humidity, could affect the measurement results?
Yes, but that's biologically interesting. We want that. So, like I said earlier, there's not a whole lot of plants that respond to air velocity, turbulence. There's a few, and my guess is that some of them are in Indonesia because they tend to be broad-leafed tropical things that grow in the understory. But all plants respond to humidity, to vapor pressure deficit. And one of the key applications of something like the 6800 is to explore that VPD response, because it helps explain things related to drought tolerance, right? And so we can take the instrument and we can put a leaf in the chamber and we can change the VPD. We can start really low where it's very wet, and then we can move to very high. And what we'll see is that if we were looking at a plot of, say, stomatal conductance versus VPD, that at low VPD, conductance would be constant, 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 and then at some point, the stomata would start to close, right? And those stomata pose a fundamental limitation to photochemistry, right? The leaf could take up a whole lot more carbon if it didn't have the inside separated from the outside environment. And understanding how a plant responds to VPD helps us understand those limitations on carbon fixation that are just purely diffusive in nature.
Okay. The last question. Oh, sorry. Oh, the next question. Okay. In general, when is the best time to use Li-Cor for measuring cereal plant?
Okay, this is a fabulous question, because it is a major topic of discussion tomorrow. So not in the context of cereal plants specifically, but in the context of all plants. And there's no right answer. There's no right time of day to go collect the data. There is a right time of day to collect the data that you're interested in. And tomorrow, I'm going to talk a little bit about how you figure that out, right? So I'm going to show some data from some measurements that we call a diurnal curve. And we're going to look at the diurnal curve, and it's going to show the carbon assimilation rate. In this case, it was measured from sunrise to sunset on a clear sunny day. And we're going to see that sort of intuitively, we might think assimilation should do this on a clear sunny day because that's what light looks like. But what we'll see is that assimilation climbs and climbs and climbs. And then when we reach the sunniest, hottest part of the day, it just tanks, it drops off. And then in the evening, it'll climb back up and then it'll start to decline again. And that dip is called a midday depression. And if I'm doing work where I'm trying to characterize the maximum capacity of something, so if I want to know the light saturated photosynthetic rate, or I want to do a light response curve because I'm going to use this in a crop model, then I probably want to make that measurement in the morning. If I want to explore stress between different treatments, then it helps me to know when that midday depression is, because that might be the best time to go collect data. So part of the discussion tomorrow will be to discuss sort of these different ways that we apply the instrument to measure these different things. And as part of that discussion, we'll talk a little bit about, okay, well, what makes sense for the right time of day to go make that particular measurement? So there's no one size fits all answer. It's very specific to the thing that you're trying to explore and the kind of data that you want to get from that.
Okay. The next question, I'm wondering about the two things. First, what are the excessiveness of the Li-Cor 6800 compared to 6400? Secondly, in previous slide on your presentation, you mentioned it about control VPD in Li-Cor 6800. If we use constant VPD in our measurement, is it mean that the temperature and humidity directly control as well? Thank you.
Okay.
Two questions.
Two things,
yes.
But they're very much the same thing. So I'm going to share my screen again. And let's see. So what we see on my screen is an image in real time of what's being displayed on my instrument console right here. And so if I touch a button here, we see things change here. So what we're seeing on the window, the screen, is one of the key differences between a 6800 and a 6400. And that is that the instrument directly controls water vapor. So on the back side of the console, there's a desiccant column and there's a humidifier column. Well, we can't see those. Let's try this. So there's a desiccant column and a humidifier column. And the instrument, as it's shown here, partitions the flow between the humidifier and the desiccant to get whatever water vapor concentration or relative humidity or VPD that I've asked for, right? And so that's very different than a 6400, which did not have any sort of water vapor control. Some of the other things that are different on a 6800 than a 6400. So I'll go to this thing called warm up and systems tests. So before we took a 6400 to the field, there was a checklist in the manual. Look at this, push this button, turn this knob to make sure that the instrument was working correctly. On the 6800, that checklist is a green start button. Instead of relying on a user to read something and interpret it correctly, perform an action correctly to decide if the instrument is working, The 6800 is self-diagnostic. We get it turned on, we put a CO2 cartridge in it, we close the chamber, we hit start, and over about 10 minutes, the instrument will tell us if there's anything wrong with it. And if there is, then it'll give us suggestions for how to fix it. And if it's not, then we can just go to the field and start making measurements. For me, those are maybe two of the biggest functional differences. There are a lot of differences that have to do with how data files are handled. So on a 6800, we have the option to copy files to a USB thumb drive. That's not something that ever existed in a 6400. There are a lot of improvements in the speed of the instrument. I talked a little bit about matching earlier, and I gave this example where you could match with the chamber open or with things that weren't stable. Well, on the 6800, that's true. On a 6400, matching was a user-driven thing, and the user had to know when it was appropriate to match and when it wasn't appropriate to match. Tomorrow, I'm going to show some auto programs and the kind of data that we collect with those. And part of that will be discussing some really key differences between a 6400 and a 6800. There's some measurements that are possible in a 6800 that have never been possible before. One of them has to do with these high-speed CO2 response measurements. But in general, those are maybe the big highlights.
Okay, Jason. The last question. Firstly, is Li-Cor can be used to monitor photosynthesis continuously for long-term measurement? And secondly, how to scale up a single leaf measurement from Li-Cor to canopy of photosynthesis? That's the question, the last question.
Yeah, so for long-term measurements, we have a couple of different ways that that's done. So one is that we go make repeated measurements on the same leaf, maybe over the course of a day or over the course of a growing season. We would call those survey measurements typically. If we do it over the course of a day, that's how we develop a diurnal curve. There are people that do this over the course of a growing season or the life of the leaf. You get information on its sort of total carbon balance. There's also long-term measurements that are made continuously on one leaf. And this is commonly done when working with cam plants. So in the first presentation, we talked a little bit about the different photosynthetic metabolisms. And I saw up there a nice diagram of cam photosynthesis. This is where in the traditional sense that the stomata are closed during the day, and then they open at night, and they take carbon up at night, and then they close during the day, and they take the carbon that they captured at night, and they use it as the substrate to do photosynthesis. If we want to measure gas exchange on a cam plant, well, we have to measure it when it's doing gas exchange. And I'm pretty lazy. I don't want to go out in the middle of the night with an instrument to make measurements. And so there are features on this instrument that allow it to run autonomously. And so we just clamp it on a leaf, we tell it what we want it to do, and it will keep making measurements on that leaf over time. These features existed in the 6400 as well. In graduate school, I used to measure cam plants with a 6400, and I would do VPD response on a cam plant. So it was one day at one vapor pressure deficit, the next day at a different vapor pressure deficit, and it took a full week to do one response curve. I did not spend 24 hours for 7 days in the lab. I would go up once a day and I would restart an auto program to let it record the data for me while I wasn't there. And we'll show auto programs tomorrow and some of the things that you can do with them. There are pre-built ones on the instrument, and then there are scriptable ones on the instrument. And then to this question of how do you go from a leaf level measurement to a canopy scale measurement, how you scale up. Generally, that's done with an appropriate physiological model. And so depending on the field, there's different models. I know that there are a number of them that are used that are crop specific, but generally it involves making measurements of at least light response at the leaf level, and making measurements of typically CO2 response and characterizing the energetic limitations. So the apparent quantum yield of photochemistry, the light saturated photosynthetic rate, saturating light intensity. It generally also involves characterizing enzymatic limitations, things like VC max and J max, and then plugging those into a model to scale up to a larger area.
Okay, Jason, sorry, we have the last question.
Okay.
Is the Li-Cor can measure fluorescent chlorophyll?
Sorry, ask that again.
Is the Li-Cor can measure fluorescent chlorophyll?
Yes. So when we talk about making a fluorescence measurement from the Li-Cor instrument, we are measuring chlorophyll A fluorescence. And specifically, we're measuring the chlorophyll A in photosystem 2. So when we talk about a fluorescence measurement, ultimately what we mean is chlorophyll fluorescence, and it's specifically chlorophyll A fluorescence.
Okay. Thank you very much for your answering for all questions.
Mm-hmm.
Maybe take a rest for a while. We are going to Mr. Professor Kukuh and Dr. Paul. Halo, Pak Paul.
Oke, Pak.
Sudah selesai seluruhnya? Apa ada yang perlu ini lagi dibahas lagi?
Kalau misalnya ada yang ingin bertanya, silakan tulis saja itu. Bisa di email atau di apapun melalui email. Nanti akan dijawab oleh Profesor Kukuh kalau yang ingin bertanya ke Profesor Kukuh. Dan juga kalau ingin bertanya kepada Jason, ya itu bisa tulis melalui email juga ke Unitama kayak gitu.
Oke. Iya, betul Pak. Ya. Jadi kita akhiri untuk sesi hari ini. Atau, sorry.
Mungkin Pak.
Ya, maaf, saya ke Jason dulu Pak. Jason, are you there?
Yes.
Yeah. Do you have anything to say or everything for today? Yeah, before
I think I'm done for today if there are no more questions for me.
Yeah, okay. Oke, Pak Paul.
Ya.
Bagaimana Pak?
Ya.
Atau
mungkin ada closing statement dari Pak Kukuh mungkin.
Oh, silakan. Silakan Pak Kukuh.
Suaranya Pak Kukuh, suara.
Pak Kukuh.
Mic-nya Pak.
Ya, terima kasih Pak Paul, Pak Memen, Mr. Jason, terima kasih. Thank you, nah, yang ingin saya katakan bahwa dengan adanya fotosintesis sistem Li-Cor 6400 ini, kita merasa nyaman dan yakin betul bahwa mengukur laju fotosintesis itu apa yang kita harapkan. Jadi ada hubungan dengan stomata, ada hubungannya dengan retention klorofil, dan juga ada hubungannya dengan hormon. Ya, apalagi dalam kondisi tercekam itu hormon EPA dan sitokinin. Jadi saya harapkan memang dengan adanya pemaparan webinar ini kita selalu ada komunikasi untuk meningkatkan kualitas penelitian kita. Itu aja Pak Memen. Terima kasih banyak.
Oke Pak. Iya. Pak Paul, kita tutup aja.
Ya, silakan Pak Memen.
Ya, kita ucapkan dulu ke orang-orang Li-Cor nih Pak. Jason. Halo Jason. Halo.
Foto bersama. Halo.
Jason. Yeah.
Yes.
We are closing today for session today, and we will meet again tomorrow. Okay, at the same time with you.
Wonderful.
Thank you very much for your session and join. Okay, see you tomorrow. Good night.
See you tomorrow.
Ya.
Okay.
Ya,
See you.
Pak Paul, Pak Profesor Kukuh, terima kasih banyak Pak.
Ya, sama-sama Pak
Memen.
Ketemu lagi besok pagi pada jam yang sama. Selamat orang Amerika Pak, maaf. Kita
Ya.
Terima kasih Pak. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.